Good morning, everyone. Welcome to On the Rise, Controls in Rare Disease Trials for Small and Diminishing Populations. My name is Garrett Homry. I'm Research Director for Regulatory Policy at Duke-Margolis. It's great to see so many longstanding friends, acquaintances, colleagues, et cetera, in the room and online. More on those exciting figures in just a moment. I was genuinely excited when I first started hearing about the formation of the Rare Disease Innovation Hub probably a year and a half ago. That excitement continued through the formal announcement, the release of the strategic plan, which I'll quote once or twice today, I'm sure. And then frankly, once we were selected at Duke-Margolis to work with the FDA team on co-hosting the first two FDA RISE convenings, I'll save news about the date, topical selection, et cetera, for when the good Amy Comstock-Rick provides some wonderful opening remarks in just a moment. Quickly realized that my excitement was shared by many. You can see that in the registration figures for today. We've got approximately 2,000 participants, either here at the National Press Club or online. You'll hear us encouraging active participation, both from folks whether you're in person or online. By comparison, we do one or more rare disease convenings each year, normally through the FDA cooperative agreements. Generally, we have about 600 or 700 participants. I think the numbers speak to a few different things. A lot of it has to do just about the excitement associated with the hub in particular, the fact that it's steered by center directors at Cedar and CBER, the fact that, well, frankly, just to quote directly from the strategic guide, that the strategic plan, that it's imperative that the internal expertise be shared within and across centers so that knowledge gained in a single IND, BLA, NDA, or during a patient-focused drug development meeting or patient listening session can be retained and utilized across the institution as institutionalized knowledge. We are all well aware of the heterogeneity across any singular rare disease, and that's a single rare disease. Extrapolate that to 10,000, maybe 16,000 rare diseases. The need to communicate across the agency, centers, review divisions, et cetera, to tackle these questions, and frankly, in concert with you all, patient advocacy groups, industry sponsors, researchers, trade associations, policy shops, et cetera. So that's essentially what we've tried to do when preparing for today. So bring together this broad range of experts, in person, online, et cetera. Learn not just from the successes, but also from the failures. Let's share openly, see what we can learn, extrapolate, build upon, et cetera. Before we begin, a couple of housekeeping notes. Actually, I don't believe I have my clicker. That's my fault. I should. If we could go to slide 3, I believe. Thank you. Duke-Margolis is convening this workshop under a cooperative agreement with the U.S. Food and Drug Administration. I'm sorry, that's my fault. As you all know, we co-host about a dozen of these each year. The conversation today, it's supported through that cooperative agreement, but it's not a federal advisory committee meeting. We will not be voting, making recommendations or other binding recommendations. That's not kind of the purpose of the venue here. But the important conversations will be had. Let's see. Oh, geez. It's always the hardest part for me, the technology. We'll begin today with opening remarks from FDA leadership. Afterwards, we'll have a panel discussion on considerations for control designs, when designing clinical trials. What are the things we're factoring in? How do we make these decisions? Who's involved in making these decisions? After a short break, we'll resume with a presentation and discussion on internal control options. Find kind of a pattern for the next couple sessions, case study, discussion, broadening to whatever the most important factors are that our esteemed contributors have been working so hard on for decades. After lunch, we'll pick back up with a presentation, with additional presentations and subsequent discussions on external control options. And then we'll end the day with a reflection of the day's discussions. Looking forward, what do we do next? Decades of work goes into this, decades of work will continue. Where do we go from here? Logistics for the day. Attendees, whether in person or virtual, are encouraged to contribute to the meeting. We tend to use the Slido Q&A function. I'll pull up a QR code in just a moment. Also, folks that are in the room, by all means, we'll be running mics. Raise your hand. We do want to incorporate you there. but I've also got a trusty iPad where we'll be shuffling questions in from you all and from folks online. Now, it's my pleasure to introduce our speakers for opening remarks. First, we'll have Dr. Vinay Prasad. Dr. Prasad serves as Director for the Center for Biologics and Research. Next, we'll welcome Dr. George Tidmarsh, the Director for the Center for Drug Evaluation and Research, so serving as the the co-leads for the hub. And lastly, the good Amy Comstock-Grick. Amy is the Associate Director for Rare Disease Strategy and the Director of Strategic Coalitions for FDA's Rare Disease Innovation Hub. My excitement was also shared when it was announced that you would be leading these efforts many months ago. But now, let's pass over to Dr. Prasad for opening remarks. Vinay.
Thank you so much for having me. I hope you can hear me well in the room. It's such a pleasure to be able to speak to you. It's such a pleasure to speak to you here from, actually, I hear the feedback from the room. You might be able to mute that for me. Okay, how's that? I still hear it to some degree. I think your microphone is on, picking up my own audio and feedback to me. Okay, thank you. So, such a pleasure to be able to speak with you today about something that's so near and dear to our heart, which is accelerating innovative treatments for people who suffer from rare diseases. And I just put together a few thoughts on this topic that I'll share with you this morning. When I took this job, I said in a number of public lectures that we at the FDA, and CBER in particular, will be firmly committed to making drug products available at the first sign of clinical promise. We're not going to wait for overall survival. We are going to make it available to patients at the first sign of promise. At the same time, we are going to continue to follow those endpoints because we also care if these drug products ultimately improve how long people live and the quality with which people experience life. And I firmly believe that conditions that are rare, dire, and have few treatment options deserve flexibility in a way beyond something that's common, that has many treatment options where people live a long time. I'm firmly committed to that principle. And in that vein, the commissioner and I will soon unveil what we call the plausible mechanism pathway. This will be a novel pathway for drug developers who are truly pursuing bespoke therapy. By that I mean completely individualized therapy, that baby A gets one treatment, baby B gets the next treatment, baby C gets a different treatment. And we will give drug makers a roadmap for how they can get marketing authorization after treating just several kids or young adults or adults with a certain condition, and they will get an authorization for a process, a platform, a pathway, and we will make that available very shortly. We're finishing up our draft. Plan to submit that to the New England Journal. We are firmly committed to the idea that randomized controlled trials are sometimes needed, particularly for common diseases that 100 million Americans may be treated for, but they're not always necessary. They don't make sense for a lot of rare conditions. And I'll give you a good example of something we recently approved. We recently approved an immunotherapy for recurrent respiratory papillomatosis. What is this? This is a condition where a handful of people suffer from recurrent, almost like warty growths in the throat that prevent them from breathing and speaking, and they undergo a series of surgical debridements for this, and they're often linked to HPV. And we have a new immunotherapy, adenoviral vector gene therapy, that is administered, that has shown in a non-randomized study that patients who get this therapy, who were previously going to see the surgeon every few months to get another resection, are now able to go even years without having to be resected again. And so this is a great example of patients serving as their own controls. This is a great example of you don't need a randomized study to know this works. This is a great example of tremendous regulatory flexibility for rare conditions, and we just made that approval last month. And I want to thank Vijay Kumar and the team at OTP for helping ushering that through. I also want to say one thing about the people who work at FDA. Now, I've been a longtime critic of the FDA. Lord knows that's true. But when I came here, I was humbled to see the hard work and dedication of the staff here. People here work from 6 a.m. in the morning. They're sending you emails before most people are even awake, and they're working till midnight, trying to accelerate regulatory actions. That blows me away. And then the other thing I'd say is I'm touched by listening to reviewers speak about what are the most impactful things that go into their decision-making? And often it is hearing from patients who are dealing with a condition, dealing with a rare disease, and trying to think about what we can do at the FDA to remove obstacles to making more therapies available for those patients. And I hear that from so many reviewers I talk to here, to the point that I'm even thinking about having a roundtable discussion so you all can hear from our own reviewers and let them talk about why they're doing this job, what motivates them, why they're working so hard, and how they want to make therapies available. The last thing I want to say. Now, we're tremendously flexible, and I think that recurrent respiratory papilloma therapy shows how we will not require randomized control trials for every condition, certainly not when things are rare, certainly not when patients can serve as their own control. But I also want to say two things that I think don't get said often enough in these meetings, which is one of the duties of the FDA is to protect people because there are two facts that are still true. Some therapies actually don't work. Now, there are some therapies that people try to bring to market, and they may have very sexy television ads, they may have sexy cartoons, but they actually don't work. And there's some common elements of things that don't work. Often the mechanism of action has almost nothing to do with the disease process. And often there's no consistent effect on endpoints. Some things seem better, some things seem worse. There's no clear period of time where you're not getting a surgery, for instance. Sometimes a manufacturer may look at 50 or 60 or 70 different endpoints, and only one or two may be positive. This is often an incorrect way to look at data. It's as if I was a novice marksman. I've never shot a gun before. I shoot at a wall and then I paint the bullseye after the fact. Of course, I look like a good shooter. And then the last thing I want to say is that some drugs actually, not only do they not help, they harm people. They result in greater mortality. They kill people. They result in harm to people. They shorten our lives. And I guess one of the duties of the FDA is we have to strike the balance. We have to allow therapies available to patients as quickly as we see promise, but we also have to be careful we're not fooling ourselves. The first person you shouldn't fool is yourself. And we don't want to let therapies to market that truly hurt more people than they help. And that's the balance that we face. That's the challenge of this job. How do you do that with a study of maybe 20 people, 30 people, 40 people? I think it's doable. I think it's doable for even studies of 5 people. And our plausible mechanism pathway will outline that scientific approach. We hope to publish that shortly. But at the same time, it's not true for every single product. So I hope you take away from this that we are on your side. You know, we're on the same team here. I hope you take away from this that sometimes what you read in the press is not exactly accurate based on we don't get to respond to everything. But I hope you feel like we will address your needs and we're planning on working with you in the rare disease space. So those are just some preliminary thoughts I put together, and I'm happy to turn it over to George.
Thanks, Vinay. Welcome, everybody. Good morning. I'm very pleased to join this inaugural Rare Disease Innovation Science and Exploration Workshop, RISE. I want to thank everybody joining us today, both in person and virtually, as we embark on this milestone meeting for the FDA Rare Disease and Innovation Hub. And I want to thank Amy for her hard work putting this together. I understand we have a large crowd. That's terrific. I think it highlights both the interest and the breadth of the interest in developing new therapies for rare diseases. This is a milestone moment in our ongoing commitment to the rare disease community, patients, academic researchers, and the regulated industry. I think we can work together to consider these new approaches, and Vinay has nicely outlined some of those, for bringing new drug and biologic therapies to allow overcoming hurdles that traditionally have impeded progress for rare disease treatments. You may know that I've spent many years in industry developing new drugs, and I've long known that CDER and FDA as a whole has built a strong foundation for rare disease programs. And I want to echo some of what Vinay said. Now, as a member of the CDER team, I can tell you I am witnessing firsthand that dedication. As Vinay said, people work extremely hard here. They're passionate, they're dedicated, and they do want to help. At the same time, as Vinay said, they want to make sure that we're not bringing more harm than good. We have to do that by maintaining our scientific rigor, and that always underpins our assessment of safety and efficacy for new drugs and biologics. To that end, I'm happy to tell you that today we're announcing a new regulatory process that stems from the often heard question from drug developers in the rare disease community. How can we ensure that rare disease programs at FDA take into consideration all types of data that are available to assess the safety and efficacy of a new treatment? Well, the Rare Disease Evidence Principles, or RDEP, I'm learning also that everything here at FDA has an acronym. This RDEP program sets forth the FDA's commitment to the community that when it is only feasible to conduct one adequate and well-controlled investigation, doesn't necessarily have to be randomized. It could be patient as their own control, as Vinay outlined. But there is other strong evidence, whether in external control, pharmacodynamic data, or expanded access data, we will ensure that this supporting data is considered in the marketing application review. So just to reiterate, we are committed to using supportive data that comes from a variety of different sources. This process is for the review of drugs to treat rare diseases with significant unmet medical need and with a known inborn genetic defect that has been identified as the major driver of the disease pathophysiology. That's a very important point. That is, we know mechanistically that there's a very strong link between what drives the disease and the therapy to correct that genetic defect. It is critical that reviews under this process are under the same rigorous scientific standards for assessing safety and efficacy of all drugs. This program offers a process that gives assurance to the community that FDA will consider supportive data as part of the application review. Importantly, sponsors may apply for this review process anytime prior to the launch of the pivotal trial of the investigative therapy, and it is specific to the inborn genetic defect in question. It can be correcting a defective gene, a mutated gene, or as a replacement of an essential pathophysiologic protein. Sponsors accepted into the program will have an initial meeting with the appropriate FDA review team to determine what data will be used to assess substantial evidence of effectiveness. FDA will work closely with these sponsors throughout the process. In the coming weeks, look for additional information on the Rare Disease Innovation Hub website regarding this program as we roll out the new process. In addition, I want to announce another important initiative that is not specific to rare disease, but hugely important to FDA's commitment to accelerate development and review of drugs. We are announcing the Commissioner's National Priority Voucher Pilot Program. Vinay and I are closely involved in this program, both selecting candidates from applicants, as well as assessing and shepherding these programs through the review process. I'm sure you've all heard about this program, but I'm very excited to discuss some key points of the program here today. The CNPV pilot program is designed to accelerate the development and review of certain drugs and biologic products that are aligned with U.S. national health priorities and to enhance the health interests of Americans without undermining FDA's gold standard scientific approval process. The voucher is non-transferable and it must be used for the development of the therapy for which the sponsor is applying. The focus of this pilot program, it will be on products that we think have a significant public health impact and can transform the US healthcare system, including how patients receive their care, the cost of their care, and focusing on the prevention of debilitating diseases to enable Americans to live longer, healthier lives. You can find complete information on the priorities on the FDA websites, the priorities of the CNPV program. This program is not designed to play favorites, but to help to close significant gaps in our healthcare system and to accelerate the development of therapies to address emerging needs. We are doing this by harnessing our internal resources and focusing those internal resources on these priorities. We have begun accepting statements of interest and have received hundreds of applications from companies who would like to pursue the voucher. We are committed to doing at least 5 in the first year and are working internally to get that set up now. Look forward soon to an announcement of the initial batch of accepted application. Between the new priority voucher program, the Commissioner's priority review voucher program, and our existing pathways, we are working to create the most efficient approval system while maintaining our scientific standards that, bar none, are the best in the world. The goal is getting more cures to the American people as quickly and safely as possible. So in closing, I know that today's meeting is the first of many that will explore topics important to the rare disease community and rare disease product development. I look to the Rare Disease Hub and others at FDA to continually engage with the community, the stakeholders, on many important issues that are critical for the patients who suffer from these debilitating diseases. We are here to listen, learn, and begin shaping a path together towards better product development. Thank you to all our speakers for sharing their expertise, and thank you all for your commitment to this important work. With that, I will turn it over to Amy Comstock-Rick, Director of Strategic Coalition Rare Disease Innovation Hub here at FDA.
Thank you, George and Vinay, for those opening remarks and announcements. And thanks to both of you for your leadership in co-chairing the Rare Disease Innovation Hub. As George said, I'm Amy Comstock-Rick, Director of Strategic Coalitions for the Rare Disease Innovation Hub, and very excited that we are here for our first RISE Research, Innovation, Science, and Exploration workshop. As George said, for the rare disease evidence principles, I encourage you to look for more information on that in the coming weeks as it is laid out. There should be information on the Rare Disease Innovation Hub actually shortly, perhaps today. I'll add my own welcome to all of you. As Garrett mentioned, as is laid out in the Hub's inaugural strategic agenda that was published earlier this year, the vision for the RISE workshops is to address challenges that are common to multiple diseases or a class of diseases, and for which evolving science offers innovative solutions. They are designed for interaction among all rare disease community stakeholders, including drug developers, patient and disease organizations, academics, FDA regulators and reviewers, and staff from other federal agencies. Today's topic is an excellent way to launch this program. This meeting will address recurring issues with controls, internal and external, for rare disease clinical trials where the disease population is already small and has diminishing numbers of eligible trial participants. Before we begin today's program, I want to take a minute to talk about the next RISE program, because this morning has clearly been a morning of announcements. First, it's important to thank everyone who submitted suggestions to the Federal Register docket for the second RISE workshop. The suggestions were thoughtful and insightful, and we are in the process of pinpointing, not just pulling suggestions for the second RISE for future RISE workshops, but also pinpointing how to share all the issues and concerns that were in the submissions more broadly within FDA. They were that good. The second RISE workshop does pull from not a single submission, but from many of the submissions, different aspects, as well as from other outreach that the Hub has had. And the second RISE workshop will be later this fall and will examine the emerging science and regulatory requirements surrounding individualized therapies, which we are defining as therapies tailored to the unique genetic variants of one or a very small number of individuals. As with today, the program will include researchers, sponsors, patients, and patient advocates to share their experience with individualized medicine approaches. Our goal during this workshop will be to further the rare disease community's understanding of and the regulatory preparedness for the evolving landscape of individualized medicine. This program will also be run by the wonderful team at Duke-Margolis, and I understand they've already begun their outreach. So if anyone from Duke-Margolis calls you, please pick up the phone or return the call. And also please mark November 20th here for that program. Turning back to today's program, I do want to thank the wonderful Duke-Margolis team for all of their outstanding work. Garrett Hamre, Christine Podsiad, and Mia Williams, who are here today. I also want to thank our speakers and those from FDA, as well as those from the rare disease community. Your insights are what make this program so valuable. And of course, I want to thank everyone at FDA who helped us plan this program. And then a special thanks to the Rare Disease Hub team, who are so committed to the goals and the importance of the Rare Disease Innovation Hub. A shout out to Kathleen Davies, Pippa Friedman, Julie Valancourt, and Nancy Gahn. You want to raise your hands? You're all here. There we go. Find them if you can. They're in the room today. And then to Stephanie Omakaro, who is participating virtually. With that, we will get the program started. And again, thank to you, all of you in the room and everyone who's participating virtually. I hope you find this day valuable, and I know I will. Thanks.
Thank you so much, Amy, and also Drs. Prasad and Tidmarsh. If I could ask for the participants in the first panel to come up, and we'll go ahead and introduce you in just a minute. While they're getting settled, we'll be very excited for follow-up announcements related to whether it was the plausible mechanism pathway, the various items that Dr. Tidmarsh spoke about all supporting data being incorporated in the determination of substantial evidence, and all of the official additional material that posts to the Hub website in the coming weeks. I believe we do have everyone. Very excited to introduce our panel for the next session. It's titled Considerations for Control Decisions. I think one thing that I don't think I mentioned in my welcoming remarks about the enthusiasm about this first meeting, there was a component about the Hub and all those things that I mentioned, but it was also central to the topic of internal and external controls and how we all try to do the difficult work of designing, executing, and analyzing data from trials when use of a placebo would produce ethical, practical, or scientific issues. So this first session, where we'll be joined on stage by Stacy Frisk, Executive Director of the Rare Disease Company Coalition, also Dr. Cara O'Neill, the Chief Science Officer and co-founder of Cure San Filippo Foundation, and then also online from Kyoto, very gracious of you, 13 hours ahead, Dr. Marshall Summer, Chief Executive Officer of Uncommon Cures, and then finally, Carmen Trupak, who serves as the Senior Director of Scientific Programs at Global Genes. Before we jump into discussion questions, can I first ask you each to introduce yourselves and share some opening remarks on what you find most critical for us to begin addressing today and moving forward. Stacy, can we start with you?
Sure, I'd be happy to. So hi, I'm Stacy Frisk. I'm the Executive Director of the Rare Disease Company Coalition, and we are 32 member companies, and each of us is dedicated to developing and delivering new treatments for people with rare diseases. And the investment on behalf of our members in this space is just really staggering. So on average, our members invest around 3/4 of their expenditures into R&D. And so this is about a $20 billion per year investment just in rare diseases. And so, the result of this is that our members have brought more than 45 therapies to patients, and they currently have more than 200 pipeline programs in development in rare diseases. And many of these would be the first FDA-approved treatment in their respective diseases. So just tremendous potential. And before this role, I worked in regulatory affairs on rare and ultra-rare diseases. So I have that perspective of the challenges of trial design at the disease level and also now the broader perspective. And, I think I just want to start by thanking FDA for being here and listening and having that spirit of collaboration and listening throughout the last few months with the CEO forums and other opportunities for us to come together and talk about these important issues. And I think in particular to the Rare Disease Innovation Hub. And I'm really honored to be here with these really esteemed panelists because it's such an important topic and how we make these control decisions. And I think Doctors Prasad and Tidmarsh set us up really well thinking about the existing standard and statute that demands us to look at what is an adequate and well-controlled investigation. And I think as Dr. Prasad described really well, we have too often in practice really seen this play out as a mandate for randomized placebo-controlled studies. But as many of us in the room know, the statute doesn't require that. What it says is we need well-controlled and adequate investigations of safety and efficacy. And the science and our analytical methods have really evolved over time where we can use, what I think we think of in our coalition as these fit-for-purpose alternatives, which we'll hear about throughout today. Things like adaptive designs and external controls, use of natural history data and real-world data, and how it's not only more ethical and feasible, going back to the title of our panel, but often more scientifically rigorous for us to use these fit-for-purpose approaches. And so, just because I'm first, everyone in this room knows I'll be very brief about the challenges that we face when we think about making these control decisions. And so, of course, these patient populations are very, very small, and often there's really significant heterogeneity patient to patient. And I think some of the diseases I've worked on, you even see that variability among two siblings who have the same disease. And so this becomes a real mathematical tension when you think about sufficiently powering a study where you're going to show a benefit using randomization. And at the same time, do we just forego development in these diseases that are challenging? No, we look for alternatives. And so, the question I think we're thinking about today on this panel is how these alternatives can actually be better sources of evidence and better positioned to demonstrate safety and effectiveness than randomization. I think one example is that, if you have a really good, well-matched external control, we have really advanced statistical methods, propensity score matching, Bayesian borrowing, where we can account for a lot of those confounding variables that Dr. Prasad, spoke to, some of the limitations. We can account for a lot of those. And actually what we can provide within a well-matched external control might be more interpretable and reproducible than what we can do in randomization, where you have that heterogeneity and there's a lot of variance and your randomized arm gets really noisy. And it may take decades, if ever, to show a statistical significant deviation in your treatment to demonstrate benefit. And so, also one last thing, as we learn more about these diseases and their underlying causes, there's a real role for the increased use of biomarkers, particularly when those biomarkers, we have a really good idea that they're able to measure the underlying biology of the disease. And in these cases, we can actually get better evidence than clinical outcomes alone. And so when we think about going back to Dr. Tidmarsh and Dr. Prasad's opening remarks and how we think about gold standard science, what we've described in these kind of fit for purpose alternatives, that is gold standard science. And as sponsors, when we think about making these control decisions, I was really encouraged to hear about this totality of evidence approach because it behooves us as sponsors to look at all of the available evidence, whether from natural history, literature, to really choose the model that has the best chance of demonstrating safety and efficacy while ensuring that all patients possible have the opportunity to benefit.
Thank you so much, Stacy. We too are honored that you are here. Era.
Yeah, thank you. I'm really happy to be part of this important discussion. And Stacy did a great job kind of overviewing, I think, things that we'll all agree on. But as Garrett said, I'm Kara O'Neill, Chief Science Officer at Cure Sanfilippo Foundation. I'm a pediatrician by training and at heart. So I come at looking at these things with that lens. But I'm also mom to a little girl who lives with a pediatric neurodegenerative metabolic disease called Sanfilippo syndrome. And that disease, children are seemingly normal at birth, and then the first few years begin to experience speech delays, cognitive regression. They go on to develop problems with their motor abilities, develop seizures, movement disorders, and require G-tube feedings before they pass away early in life. There are no approved treatments for this disease. We've known about it since the early 60s when it was first described. And as all of you in the room can appreciate, time is always working against us in this progressive disorder. And that's why, randomized control trials present a really extraordinary challenge, making them both unethical and infeasible for many of the contexts that we're talking about. I think, I want to focus on the neurodegenerative space because that's where I spend my time. And thinking about the context of child development. Neurodevelopment is a time-sensitive process. It's not like many of the other kinds of conditions or common conditions that maybe you can rescue a situation later down the road. Maybe you have time to find out what works, what doesn't work. But neurodevelopment happens at a very critical point in life. And if you miss that window, you will irreversibly damage the child's brain and future abilities. So that's a fact. There's no question about that. And we need to think about toxin exposure. And I know that's something that, I'm glad has come to the forefront of our thinking here in the US and thinking about the things that we're exposed to and that we're exposing our children to. Things like lead exposure, we know causes intellectual disability. Elevated phenylalanine levels in people with PKU cause severe intellectual disability. Likewise, elevated heparan sulfate levels in people with MPS diseases causes severe cognitive impairment. It's not different, it's the same, but we still do not have any treatments for the neurologic impacts of MPS disease. And so we have to ask, why is that? We understand the biomarker, it's well described, we have evolved in our ways to measure that in very specific ways. We would never purposely expose our kids to toxins. So, when we think about a randomized trial, we have to take a step back and be pretty concerned when we do still hear regulatory advice that is advising the use of randomized control trials to demonstrate cognitive endpoints in neurologic MPS disorders. I'm really encouraged by what we're hearing on the screen today, but we need to have that implemented in practice. And now, because we are losing therapies on a regular basis and over the last decade, and our children still have no treatment. Thinking differently about evidence generation, I think that's why we're all here and are going to hear some really wonderful ideas about how to advance that. But use of the natural history data from various sources and these validated primary disease-causing biomarkers can and should serve as meaningful comparators. Equally, we need that flexibility to have accelerated approval based on those biomarkers and then see that clinical evidence bear out because these symptoms evolve over maybe a decade or more. And, industry can't sustain trials for that long in these ultra-rare diseases. So again, I'm really encouraged by the focus of this meeting, by the existence of the Rare Disease Hub, by the things we've heard today, and hope that we can move towards embracing these new trial designs, not new, but apply trial designs that both respect the humanity of our children and balanced with scientific rigor.
Thank you, Kara. Carmen.
Didn't expect to go next.
I have to admit, I felt a little awkward jumping to Marshall, and it might be exclusionary to you. It was a debate that I was going through in my mind.
Yeah. Yep, I'm Carmen Trupek. As Garrett said, I direct scientific programming at Global Genes. Global Genes is an umbrella nonprofit organization that supports hundreds of patient advocacy organizations. And in the work that we do, a lot of that work is really supporting these organizations and the patient advocacy group leaders at those organizations in developing and carrying out some of their research strategy. We call these patient advocates next generation advocates. And that's really a reflection of the work that patient advocates are doing today. Patient advocates today aren't just raising money and handing it over to support research. They're really driving a lot of the research themselves. And something I want to take my few minutes to talk about today is the development of some of these data sets and really what that takes from the patient communities. So in particular, many of the organizations that we work with have registries. They're really working on collecting data through those registries, but almost all of them come to this almost crisis point about whether or not to conduct a natural history study and whether or not to really like put all of their resources, and it will take almost all of their resources, into a robust natural history study. And conducting a natural history study in the sort of gold standard way, when we hear that term, really requires a number of things that are very rigorous and sometimes quite difficult to achieve. Number one, they should be prospectively designed, intentionally designed with a protocol. They should be longitudinal in nature to collect longitudinal data in that very standardized, structured way. They should include standardized assessments, ideally ones that can be repeated in a clinical trial environment. And then patient-level de-identified data should be made available to researchers conducting those studies. That's a tall order of 4. That's a big order. It's extraordinarily expensive. It takes a long, long time to do, and it's hard on families. There are a number of estimates that suggest that probably 1/3 of all patients with rare diseases who enroll in natural history studies never return for a second visit. Drop-off continues after that. So it's because they're hard. You have to travel. If you have a child with a neurodevelopmental disease, a 7-hour evaluation is extraordinarily difficult. And so what can we do to change that? One thing that excites me beyond today has been exciting already in some of the comments and thinking about the way that the FDA is clearly looking at different types of data and the way that we use different types of data in this sort of totality of evidence. But also one thing that's exciting to me is if you look at historically studies where the FDA has approved drugs based on external control data, what you see is that the vast majority of them do not actually use natural history study that's collected in the way I just described, right? Most of them actually use a combination of data, and the most common type of data that gets used is retrospective natural history data of some kind. The difficulty is predicting when that will and won't be accepted. And the vast majority of conditions where that has been accepted to date have been conditions that are very severe and where the treatment effect is very large. So if you think about the classic example of SMA, the outcome is survival. Children who died and children who now live a very happy life. Dr. Prasad mentioned this rare respiratory condition, RRP, that was just approved last month, very exciting. But the treatment effect was complete resolution and no longer needing to have resections, right? It's complete resolution. So we know that this is not going to be the case for most rare diseases. It's not. We are not going to see that large of a treatment effect, especially when you start talking about the thousands of neurodevelopmental disorders where progression is very slow. So I think the patient community needs guidance. They need to understand what types of natural history studies will be accepted and for what types of diseases. I think we need to work together on protocol designs that enable the kind of things that we already know keep people in trials, which are non-traditional designs that enable people to participate primarily from home, where some of these standardized assessments can be done in the context of their own medical care and the providers they know. Combinations of retrospective EMR data review plus prospective assessments. I'm really encouraged that there's lots of opportunity to do that together.
Thank you, Carmen. Marshall.
Well, first off, greetings from Kyoto. Is the audio coming through okay?
Brilliantly.
Excellent. First off, my name is Marshall Summer. I've been in the rare disease field for around 40 plus years at this point in time. I've actually had the benefit of watching from the early days after the Orphan Drug Act passed until the modern days, the incredible growth in the field, the number of studies that have come through, all of the really kind of good science that's come through. And I've also had a chance to watch some of the shortcomings and shortfalls we've hit along the way as we're learning about these things. I've been part of somewhere north of 150 rare disease clinical trials over the years, whether as a PI or a peon. In that standpoint, we've gotten to see a lot of things. One thing that kind of we have to keep in mind is actually the simple math of rare disease. If you look at actual incidence and prevalence of rare disease, we're probably coming up somewhere between 14 and 16,000 rare diseases. And as we discover new ones, the number of patients in each disease goes down dramatically. The sort of common ones were already kind of known, but last time when we kind of ran the numbers on this, probably about 90% of rare diseases have fewer than 150 patients. And probably about 80% may have fewer than 50 patients. So there's a reality of the available number of patients as you start looking at a lot of the newer described diseases, but even some of the older ones that we know about, that you have to take into account when you're designing rare disease clinical trials. And then if you look at the failures, if you look at, kind of the overall success rates, the numbers tend to hover historically around 25% success rate for rare disease, which is actually the best kind of drug trials coming through from phase 1 to phase 4. But when you look at the reasons for failure, it's not, the science is not the major one. That's maybe 20%, 15%, somewhere in there. But there's a couple of good studies that show that 55 to 60% of rare disease trials fail because of recruitment and retention. And what that tells you is whether it's the design, the available number of patients, the study burden of the disease, all of those different factors that come into play there are actually causing us to not see treatments that we would otherwise maybe have for our patients. And then, of course, the old gold standard. Gold standards are funny how they get to be, history sometimes anoints them, whether the science anoints them or not. Was the rare, double-blind placebo-controlled trial. But the number of patients you need to detect a signal there is actually quite large. And sometimes just to even pick up an effect size of 0.5 or something along those ranges, you may need more than 150 patients, which is maybe more patients than are available for any given rare disease. So you really are forced just by the realities of the size of these patient groups to really look at novel, and not even novel, but just different methodologies for that. And I've noticed that we've got a deep statistical toolbox now, but we don't really go too deep in it for a lot of the tools because I think it gets out of people's comfort zones and people kind of like things that have worked before. So if it worked once, we kind of want to use that particular tool again. The only other thing I'll mention, like I said, it's hard to go forth in a group like this because most everything gets said, is genetic heterogeneity. The majority of rare diseases do tend to be genetic in origin. However, just because you've got a mutation in a gene doesn't mean all patients look the same. I've worked in urea cycle disorders for most of my life, and my youngest patient presented at 4 to 6 hours, and a patient with a defect in the same gene presented at about 80 years of age. So there's a big spread there in the presentation, severity of disease, impact of environment, things like that, where when we try to do your classic group comparisons, where you try to lump, patients into a control group and a treatment arm, you may actually miss a lot of things just because there's so few patients, and a few patients with a big difference in presentation can really throw things off. So I'll kind of close up there so we can get into the discussion here, but it's an honor to be here and appreciate being asked.
Appreciate you all very much. It is very helpful to have the level of precision and elegance where you describe the challenges, the stakes, but also some of the technological opportunities, some of the lessons that we've learned where we have had successes. And as we talked about in the beginning, as much of this work is about building off the failures, not the successes. So I appreciate the clear description of the numerous challenges that do exist. Before we dive into some of the more kind of technical questions, can we stick with a minute? And I think I'll start first with Kara and Marshall with this one. Can we talk a bit more about some of the key ethical and feasible considerations that patients and families go through when considering trial design for small and diminishing populations, when considering this, 90% of rare diseases might be fewer than 150 people? Kara, can you talk a bit about who should be at the table when defining acceptable risk? What are just some of the overall ethical and feasibility considerations that come to mind to you? And then I'll go to Marshall next.
Yeah, thank you for the question. I think we'd all agree that patients or people with the lived experience need to be at the table. And what does at the table mean? I think we appreciate that FDA wants to be in listening mode a lot of time, but one-way communication is not very effective. And there needs to be opportunity for actual conversation, not just listening, because I think that's where we really can understand one another. Patient advocates need to be in regulatory meetings as a norm, not as something that is when there's a problem or when there's a serious concern. We should be able to have that transparency and that collaboration throughout the development process. And, I recognize that there are potential barriers there around confidentiality or things like that. But, what can the FDA do to encourage sponsors to bring an advocate? Or what can the FDA do to help bring an advocate in on their side so that everyone benefits from having the full perspective represented in these discussions? Because, one decision can really take a drug development path down a different road, and it might not be the best road for the patient community, who are the ultimate people who need to be accepting of the drug.
Marshall.
So a couple of good issues to bring up here, Kara. Those were some great comments there. Obviously, the patients and families need to be at the table here, but there's actually kind of another interesting thing, which is when we start talking about risk-benefit rating. I have a lot of friends at FDA on different panels and things, and they have a tough job because on the one hand, we always yell at them faster, faster, we need these now. But if something goes wrong, or say a drug proves to be unsafe or something, sometimes they're the first ones on the bus. So how do you actually instill that proper appreciation of the risk to the patient, but also the potential benefit and the family's willing to accept, or the patients being willing to accept greater risk from that standpoint? I think it's something we probably need to be a little more open about, because from a regulatory standpoint, sometimes you want to go with the more conservative thing. But for patients with degenerative rare disease, that in some ways actually makes the higher risk situation. If you're insisting on, placebo-controlled study, which is going to take 10 to 15 years to recruit, there are going to be a number of patients who will die during that study just because, and for a drug that would potentially be effective, just because you have taken that more conservative pathway. Now, at the same time, and I think there's some great points made earlier, we want to make sure that the drugs that come through are actually working and doing their job. But I think the families and actually the clinicians who work with these patients on a regular basis really need to be at the table, and not just there as it's nice you're here, but actually as an integral voice on what is going on in these decision-making processes. And then you have to really look hard at these things on what type of risk are we willing to accept for these things? What type of risk are we willing to accept we might be wrong? Because sometimes you're so afraid of being wrong, you're scared to be right.
Yeah, acknowledging that there's harm in doing nothing. There's very much harm in doing.
And I've seen that happen a number of studies where, either they were wanting a placebo arm or the number of patients required just could not be done in a reasonable amount of time. And while waiting to do those things, a therapy that would have benefited patients wasn't available. And a number of patients died. But we don't ever really do that calculus. And I think that's something we actually do have to start accounting for.
Stacy, can I ask you something, a similar question, but tilted a bit towards you have this unique experience of having very much of the kind of company investment line of sight because of your organization. You talked about 3/4 of investments and where those go, et cetera. At what point would you say requiring a traditional control model risks a trial, being feasible at all for companies specifically? How do you help them make these decisions? How do they make these decisions?
Sure. And first, I just wanted to add on too that, you did a great job demonstrating what's at stake for patients when we don't get this right. But I think also, if we fail to initiate a trial or it's not a successful trial because we failed to consider these factors that my fellow panelists raise, of course that impacts the patients, but there's also the downstream impact of how this impacts how researchers, sponsors, and investors look at these diseases, and they may think, well, there's no viable path forward here in this disease, and so that limits hope for future treatments for patients in that disease. And so we have to really seriously consider the harm that we cause by doing nothing, I would say. And back to your original question, which I will answer. So, I think it's not always as straightforward because it's really simple when we think about diseases that impact very, very, very few numbers of patients. And there are some great examples where we've really leaned in and been able to find novel solutions in those contexts. But when we think about what is the disease archetype that really challenges our traditional way of doing things, it's not always that straightforward because you could have a disease that has a relatively higher prevalence but still being a rare disease. But there are other variables. Maybe there is, significant heterogeneity patient to patient. Maybe it progresses a little bit more slowly over time as compared to the SMA example, which is a very common comparison that makes it very challenging. And I think it's also really important to note here that when we talk to patients, we talk about clinically slowly progressing, but for these patients, the need is really urgent. They are not slowly progressing. Every single day is lost function. Every single day is organ damage. And in many of these diseases, that progression is irreversible. Once it's done, we can't get that function back. So I think we need to keep that in mind when we're thinking about these diseases. And one other important point I want to make, and I think, Dr. Prasad, really encouraging remarks about the role of how we look at evidence in the context of these rare diseases when they're investigational. But it's really important that we consider that these same challenges that we're talking about today, they exist in the post-approval setting. So when we have accelerated approval, for example, based on a surrogate endpoint that's reasonably likely to predict a clinical benefit, those same challenges, the rarity, the heterogeneity, the difficulty in showing statistical significance of benefit, those same challenges exist in the post-approval setting. And in fact, they're compounded because we're asking patients to potentially participate in a long duration placebo, because again, we're talking, it could take decades for us to measure a clinical benefit in a slowly progressive disease. So we're asking patients who have an available therapy on the market now to forgo that therapy to participate in a placebo-controlled study. It just isn't going to be feasible. So we need new ways of thinking about the post-approval confirmatory evidence as well.
Thank you very
Eric, can I
much.
throw something in on? Can I throw
Please,
something in on too?
Absolutely.
We're seeing more and more in the clinical trials field, either patient organizations funding their own therapies. We see a lot, of course, the companies that bring these therapies forward are kind of small companies with limited resources. And for many of these diseases, they kind of get one shot on goal.
Mhm.
So it's not like we're going to get another statin or we're going to get another blood pressure medication when there are a lot on the market already. So we have to remember too that these trials are they're precious, and you don't often get a chance to go back and do it right if you don't get it right on the front end. And that has both a patient cost and it also has an economic cost, because it then discourages, just as Stacy said and others said, for other folks from coming in and trying again in that field.
Appreciate it very much. And also the call out to the post-market needs. Okay, well, maybe stick with communication with one more, and then I've got a few technical things that I want to get into with you all. We have heard for ages, we need to communicate early and often, early and often. It's patients need to communicate with sponsors early and often. Companies need to communicate with the FDA early and often. Carmen, can I start with you on the patient community to company side of things? Because it's an important concept, but I think we often lack the granularity. When? How early? What specifically are we asking? How often? What do we mean by early and often? Can you share some of the research you all have done on communication between patient communities and companies where that's been successful, where we can learn lessons, et cetera? And Stacey, I'm going to end up going to you next with more of the kind of company to FDA early and often. What does that mean? How? Level of granularity, et cetera, et cetera.
Yep, happy to. So, yeah, we have done some work on this in collaboration with corporate partners in the rare disease space. Because I've even heard some people say, like, it's so cringy early and often. I've heard it so many times. You've heard it too, Marshall. But the reality is that it's just a nice kind of way to think about how important it is really to have this ongoing communication. I think in the deep dive interviews that we did with patient communities and with lots of corporate partners who've been in the rare disease space, what we found in looking at sort of some successes and not overt failures, but misses, we'll say, is that companies today are very, very good, by and large, at engaging patients, the patient community, toward the end of drug development. They're very good at it. But it's the early piece that is a really critical piece, because what has happened in many drug development programs is you get to the point of a really critical trial, whether it's a true pivotal trial or not, a really critical trial for that community. And maybe there's an endpoint that's been developed that doesn't really adequately assess what's important to the patient community and what's going to show meaningful change. Or maybe the therapy has been developed, honestly, against the wrong symptom entirely for that patient population. And so getting involved very early on to understand this is critical. I started my career in clinic, and I was involved at one point in a clinical trial program for a rare retinal disease called achromatopsia that probably no one in this room has ever heard of. But Marshall has, of course. So this is a condition where children are born with almost no cone cells. And because of that, they have very poor visual acuity. They have almost no color vision, but they also have exquisite light sensitivity that's extremely debilitating. But in the early stages, all throughout preclinical development, everything was focused on visual acuity. And enrolling for that clinical trial was almost impossible, because what the patient community felt was, well, there's risk. This is like a subretinal injection. There's risk to my vision and of course it's unproven. That's what the clinical trial's for. But all of the preclinical work had been focused on visual acuity. And these people told us time and time again, like, I'm well adjusted to my vision, but the light sensitivity is so painful. It's so difficult. It prevents me from doing all kinds of social things. And if the preclinical work showed that will clearly change, or there's a very high likelihood that would change, I would probably enroll, but I won't. So that's stuck with me for many, many years. It's such a good example of how the patient community, really was not broadly engaged early on. That program was eventually dropped, and in part because they did not meet endpoint.
Mm-hmm.
But also I think that it was just so difficult to engage the patient population.
Well, it's an instructive example. It also ties to what Kara was saying before about, and Marshall, I feel like you've used this term before to me too, this expression of like, the patient voice can't be tokenized, like a dialogue needs to occur, needs to be heard, et cetera. Okay, helpful. Stacy, anything on more sort of the company side of things, adding more granularity to the early and often communications? Communicate with who, when, how, about what?
Sure. So, and I will agree, early and often, we see that a lot, and it's great advice. But you know, what that really means, I won't go into kind of, you know, the bits and pieces about where and when and how. I think FDA does a great job. There's a great guidance that has all of the available meeting types. So if you're a sponsor, you know, I recommend you look at that. I'm happy to talk with you, and there's lots of experts in the room. And I think FDA, in particular, the Rare Disease Innovation Hub, does a great job of talking about all the ways that sponsors can interact with FDA, and also, you know, recommendations for how they should include patient perspectives, again, prospectively, because that's very important. But one thing I'd like to talk about when I think about early and often, you know, is really just that it's on us as sponsors to recognize that often we are approaching FDA with perhaps novel solutions to challenges that are also novel in diseases where we might have no precedent or very little precedent. So I think the important thing for all of us to keep in mind is that it really requires us to have a mutual, that's FDA and sponsors and patients, mutual understanding that we are going to learn more about the disease and learn more about our product over time. And so our approach to development needs to evolve with those learnings. For example, if our natural history data set matures and we learn more, you know, we need to be positioned to adapt and have the mindset that we accept that it's probably a reality, not a failure, that we're adapting as we go along development, and that our expectations are also really phase appropriate. And when we fail to do that, what we really do is risk losing the potential of that treatment, and again, you know, squashing innovation in that given disease. And I think one final point, you know, Carmen did a really great job overviewing, you know, the real value in engaging patients really early and prospectively, but we also have to have an appreciation that this is a burden to patients in some circumstances to participate. And so if we apply the same rigorous or unrealistic expectations to our development of these data sets, you know, in collecting COAs and PROs, we just may never get there. So we need to not only be flexible, and really, it's not flexible. Like, these are appropriate methods, right? And they're scientifically appropriate. But we also need to be fit for purpose in how we develop that evidence.
Garrett,
Garrett, can I make a comment? Sorry.
go ahead, Marshall.
Kara first and then Marshall.
Yeah, Kara first. Yes.
Okay. I just, it might be helpful to hear a success story. I think of where, you know, we were able to kind of respond to what we were seeing, which was a very limited efficacy outcomes that were being advised and implemented in clinical trials solely around cognition. And understanding that that then limited the treatable population to a very young population in order to be able to demonstrate that, you know, significant treatment benefit. And we needed to think ourselves, help the FDA, help sponsors understand, well, what would they measure in the whole population? Because just because a child has experienced cognitive impairment doesn't mean they should be shut out the door from treatment. I mean, that's craziness. That's what's been happening, though, to be honest. And so we conducted a caregiver preference study with a series of experiments, prospectively designed, and met with the FDA, and they were very gracious. CBER and CDER came together and met with us around the design of that study, because when we conducted it, we wanted them to feel confident that the outcomes were going to be useful in their decision-making. And so we did that and then came back to them with the results and met again. And, that was a really useful interaction, and I think provided a basis for sponsors to then start thinking about, how else they were going to measure, not only in the trial population, but in these long-term post-approval marketing studies where we know that those outcomes are going to take longer.
So a great example, and I also appreciate the emphasis on success. I remember a time 15 years ago when there were virtually no orphan drug approvals or rare disease approvals, and I feel like it evolved to a point where it might have been half of the approvals over the last many years. So I do want to make sure that there's a cognizance that is as high and difficult as the stakes are in the 10,000 rare diseases, et cetera. There have been some extraordinary things that I've seen over the years. So yes, please let us build on those as well. Marshall, you were going to say something.
Yeah, what I was going to say is there's I think an underappreciated meeting type that I kind of wish people would use more with some of the sponsors we work with, which is the interact meeting. So many of the FDA meetings are kind of more formal. The answers, you're sort of locked in and obligated. And I think that leads to a much more conservative approach when you're talking to the FDA about things. You want to bring them something that you think they will accept. The thing I like about the interact meeting, which it's, the pre-IND, it's before the pre-IND meeting, is that it is actually an open, non-binding discussion. So I found that FDA folks tend to be more open in those discussions there. I find that sponsors don't use it enough, but it's a chance where you can float different ideas. Well, we're thinking about this. What about this? What about this direction? What do you think's likely to help us out? And it's something that's evolved too. A lot of it used to be around, CMC issues, things like that, particularly at Cedar, but I think it's evolved now into a meeting where you can come in, talk about study design, talk about, okay, here's the disease. Here's what we think we can do with this. Here's some realistic measures that might be available. Let's talk about how we're going to use those to get an effective trial. And I think it's something we should be using a lot more in the rare disease world.
I appreciate the specific call out to the interact meetings. Marshall, I'm going to stick with you for the next question, but also want to remind the folks online, our team just recirculated the through the Zoom chat, the Slido link, so folks can submit questions online and they get patched to me. Also in the room, we have the opportunity. So I think we're going to have somebody in the back that will have an audience question in just a minute. Definitely want this to be interactive with you all as well. Marshall, sticking with you, something kind of technical, but Dr. Prasad brought it up. Other folks here I know are interested, and we might have other folks on the panel to address it also. So given the heterogeneity of most rare diseases, where have you seen patient data as its own control, producing at times far more robust data than the average from control treatment groups with a few patients that can wildly skew? So patient data as its own control. Can you offer insights there, and certainly welcome it from others too?
Certainly, actually, one of the reasons I'm Kyoto is we're presenting an abstract on a study where we're using patient as their own control in a proof of concept in urea cycle. And let's start with what does the mean by patient heterogeneity. So we'll take a biochemical disorder where you've got the disease is all defects in the same gene, but some may take it down to only, 80, 70%. Some may take it down to 0% function. And so you're going to have really different biochemical markers. Even if you've got a really good biomarker, the use of that, if you try to norm it across the patients or average it, is going to make it wildly inaccurate because you're going to get very large standard deviations because each patient's going to present very differently. What we find is if you take the trend for each patient based on where their biomarker is or where their disease is, the signal becomes much more powerful. You can use things like linear mixed model analysis, a number of different things where you can look at what's the slope of that patient's, and the slopes are all heading the right way. That actually makes it a very powerful model to use patient as own control. Also, I think one of the things we're going to have to start looking at is patients are at different parts of their disease. So this idea that one size fits all or an outcome marker is really we're gonna have to look at that from where they are. They're very early on. It may be the biomarker if it's later on, where there's a progressive symptom coming on. We're gonna have to look at some mix and matching for some of these patients, which I know makes all the biostatisticians in the audience just cringed. Probably, gonna have to go take some antacids because that's just kind of almost a heretical way to put it. But we really do need to start looking at those things. But the biggest thing that happens, particularly in these, as the group gets smaller, a patient who's got a variant or a wilder swing on their disease can really skew and mask the signal for the rest of the group. So by breaking those patients down into each one as their own, patient is own control. I think that makes a really powerful thing. Janet Woodcock and I are currently working on a paper on this that we'll have out later this fall, looking at the different models and sort of what you can and can't pick up.
Thank you so much. I very much look forward to returning to this topic pretty extensively in other forums. Did I see a hand raised for a question? If not, there's no pressure. I just may have been mistaken. Oh, yes, I thought so.
Hi, my name's Layla Cooper Smith. Thank you for all of this wonderful information. What's your, just to go to step back, and I know you already mentioned it might be too much on the, the feed, on the communication of the communicating with the patients. I'm sorry, I forget what you called it, but the feedback loop, if you could give an example of success, what you found with success on the feedback loop with the patient advocacy group during development for the early and often. That's what it was, the early and often. Where have you seen success? Can you describe a success or a few successes with the feedback loop for early and often?
Marshall wants to do it. He's raising his hand.
So I've seen two things. I've seen one where a failed trial, then the sponsor went back and actually had a lot of very interactive discussions with FDA, presented the problems there, and by taking that approach, actually got a much better study design that then is going forward. It was able to, looks like it's actually starting to work. I can't mention the specific here. But I would say another example would be a case where a sponsor was bringing a new drug for disease, and they thought, oh, we're going to have to do a whole bunch of animal testing in this. It was similar to another drug out there, kind of a chemical derivative from that. But by meeting early and often, the FDA said, no, you don't actually need to do these steps. You can go straight to this clinical part of that. Probably saved them a year or two on getting this drug into the trial and getting the patients moving forward on that. So I've seen a couple of really good examples. Some of these were out of Interact, others were out of some of the more structured FDA meetings. So we do throw the phrase around a lot. I have actually seen it, though, have impact. And I think the FDA likes to engage. Most of the folks I know at FDA are scientists or clinicians. They want to bring their brain power to the table and help you try to get as good a trial design as you can. And also make sure you don't fall into some pits by doing some things that aren't going to work. So for me, I've seen actually really positive impact there.
In a little bit more around early and often with the patients, with the patient advocacy groups, in order to, and the sponsors, so patient advocacy groups and the sponsors, the early and often there, and the feedback loop back to the patients, but also the time to change, to implement changes from the patient feedback to the sponsor, that type of feedback loop for early and often, which I don't see enough, I feel like.
Carmen, I
But
think
if
is
there's
gonna, I'm gonna hand over to Carmen, and I'd also almost go back to, we convened a meeting, one room over, January 31st, 2024 on quality by design and risk-based monitoring, and how do you basically design trials properly from the start. And we had 2 case study presentations from patient groups, one from the CF Foundation, very excited to have their participation later on today as well, and also from Michael J. Fox, Parkinson's, I believe that was the second example. And there's some really good case studies there. All of that material is public, like all these recordings and presentations today. But Carmen, please.
Yep, sure. So yeah, I think I'm going to give you an example, and I'm not going to be able to give you the disease or the sponsor because it's currently ongoing work, but we're working with a sponsor and a patient advocacy group right now who've actually had ongoing interactions for a number of years. And the sponsor has a repurposed drug. This is a neurodevelopmental disorder that also has multisystemic organ problems, not full failure, but dysfunction. And so this presents this issue, and going back a little bit to what you were talking about earlier, that if you have a neurodevelopmental disorder and your drug doesn't focus on cognition, is it impactful? And so they had some early conversations with the community to try to understand this. Like, would this drug that really does look like it's going to address some of these other multi-organ dysfunctions be really impactful in this patient community if it does not impact cognition? And they got resounding yeses from those interactions. They brought that to the FDA. That's really propelled them. Now they're at the point where they need to formalize that into some data to bring forward, and not this series of kind of informal conversations. So we're actually conducting a burden of illness study in this community to get sort of a wider swath of the patient population and get this data to bring forward into a more formal meeting with the FDA and all three parties.
Appreciate that.
Those
example.
kind of studies are just so critical for, your foundational work. I have to call out Annie Kennedy, who was my shepherd in that process early on. But I also was able to participate in an Interact meeting recently, and just to kind of share, simply being in the room allows conversation questions to be answered in real time, to get that feedback, and it changes the thinking. I don't want to say changes, but it makes the thinking more relevant to what needs to happen, and everyone can step forward together more confidently and in the right direction. So
Anything we can do to better publicize, amplify, make these examples available to others is going to be such a critical marker of success for the hub. I think I get to have so many individual conversations. Our team does also, but it's sometimes difficult to get it recorded, public, written, shared, et cetera. So appreciate them happening here. Okay, we've got 6.5 minutes, so I think it's going to be time for one more question. We have hand raised in the front. Before you ask your question, I want to tee up something that I don't think we're going to get to now, but like moderators, other people in the room, to put this in the space. One thing I don't think we're going to get to now is the thought of whether FDA, and maybe even in concert with EMA, could consider using control model archetypes, right? So think of a few different rare disease categories: rapidly fatal pediatric versus slowly progressive metabolic versus relapsing disorders versus irreversible treatment change. Might archetypes be an opportunity that we don't necessarily have a lot of dialogue, guidance, et cetera? But that's not for now. Please.
Yeah, I want to follow up a little bit on who's at the table and what patient advocates do at meetings, regulatory meetings. For those of us who run meetings, and at least for myself, I'm struggling to understand what's the role and how they would help. So I heard about the Interact. So you sit at the Interact. So that's normally a very informal meeting. Perhaps we're discussing high-level design. But you generally will go to meetings like after a phase, like, well, in rare, there are no phases because it's like continuous. But after maybe after the point where we have to make major decisions and we're going back to the agency, can you help us visualize what that looks like? Like, what will you do at these meetings? Because generally they're very scientific, driven. We're discussing the design, the endpoints, perhaps we're tweaking. Yeah, you can bring the voice of the patient there, but I just don't comprehend.
Maybe there's this misperception that the patient community isn't educated or professional enough to sit in those rooms, and that's just not the case. I think there's learning. Nobody kind of is born with understanding regulatory processes, so I think there's opportunities to learn. But we bring insight that no one else in the room is able to have, honestly. And whether or not they need to speak to a certain issue will depend on the specifics of the meeting, of course. But being able to be there as a sounding board for questions that come up, I think is invaluable just alone. And it also brings the spirit of transparency and collaboration to the drug development space, which I think is really important and an opportunity for all of us to grow in.
Sure, and if I'm able, I can add two areas where I think it's so helpful. And your question is particularly around having patient voices and engagements with the FDA. And I think it's so important to contextualize the disease, particularly when we're selecting endpoints. It's one of the most challenging things we'll do as sponsors together with patients and the FDA. And a lot of these endpoints that we have, it's really difficult to know what a couple point change means on a composite measure that has maybe 30 measures that you're asking patients to complete. What does that mean when what matters to patient is really the activities of daily living? And so having that perspective in the room to not only contextualize, but also really help quantify for regulators and sponsors to understand how we map those things we look at with our endpoints to the things that patients actually care about. And then I think the other thing that's really important is when we look at what is acceptable benefit. I think we talked a lot today about the expectation for a very large effect size. And as Marshall very well said, we've, for lack of a better term, picked a lot of the low-hanging fruit, and a lot of the diseases we're working in, we may not see that expected effect size in a very short time frame. And so understanding what actually matters to patients is very important, because often, and in most cases, if you have a progressive disease, slowing progression or halting progression, that is the definition of a meaningful benefit. And I think patients in the room can really help us all be grounded in that reality.
Okay, we're going to wrap up this session now. Oh, Marshall, I could see that mouth opening. Are you about to speak to something?
I was just going to throw in a pitch for the archetypes. That was kind of a concept we've been playing around with very quickly. I think the nice thing about FDA and EMA coming up with sort of here's the playing field for this type of rare disease, progressive, acute, progressive, degenerative, things like that, will provide better starting points for both patients and sponsors when they're looking at, okay, is this where you would want to use a control group? Is this where you want to try to avoid a control group, since our topic is control groups today? And I think that's something that really does need to be explored. So do pass that on to the other moderators.
Can I add one thing to that?
Absolutely.
Marshall loves this phrase of the archetypes, and I think that's a good descriptor. I think a lot of people think about this as cross-disorder research and approach, right? It's a similar kind of concept. There are over 10,000 rare diseases. We can't come up with strategies and protocols one at a time. We need pathways, and those pathways are going to be different based on different diseases. So when you have a neurodegenerative condition for which children universally die before the age of 5, you're probably going to have a different set of kind of standards for what that control data set needs to look at like, versus what you would in a much more slowly progressive condition. And so it's not rocket science to group disorders into what's similar in terms of symptoms and symptom progression. There's a lot of opportunity, I think, for work there.
Sincere thanks to the 4 of you. Those of you in the audience. Also, I did start getting some questions right at the end through Slido. It looks like one of them was answered pretty well, so that's helpful. But I just want to flag for folks online. I did see that. It just kind of came in a bit late. So please keep that coming. We would like to have your online participation also through the rest of the day. We are taking a 15-minute break, and we will be back with our session on internal controls.
Well, great. Welcome everyone to session 2, which is going to further explore the issue around all the challenges and considerations associated with selecting internal controls. Obviously, this afternoon, we will get into a panel that focuses more on external controls. I'm Rachel Schur. I'm an FDA regulatory lawyer at Manat Health and really excited to be here and be part of this incredible meeting. I want to thank FDA for its leadership and perseverance in continuing with these meetings. These are just already have an incredible contribution to the conversation around the numerous and evolving and persistent challenges in rare disease drug development. I also want to thank the Duke-Margolis team, who I have witnessed over the last several months has been hard at work, very much a labor of love to get this meeting on the books and off the ground here. So thank you so much for all of your efforts, Duke-Margolis. This panel is going to function a little bit differently. We're going to have a speaker come up to give an internal control case study presentation. And then after this presentation, I'm going to invite the entire panel up to discuss the presentation and other issues that we want to cover today. So without further ado, I'd like to invite our first speaker, who is Todd Paparello, Vice President and Global Head of Regulatory Affairs for the Specialty Care Global Business Unit at Sanofi. Todd.
Thank you for the warm introduction. It's a pleasure to be here to share the case study on use of intrapatient comparisons in severe hemophilia A. Perfect. Thank you. Technology might not be my friend either. So for those of you not familiar with hemophilia A, it is an inherited bleeding disorder. It's carried on the X chromosome. It primarily affects males, and it's characterized by a deficiency of the coagulation factor 8. When that factor 8 is deficient or it's dysfunctional, the rest of the coagulation cascade cannot be appropriately activated, ultimately resulting in lack of clot formation and bleeding. And so as you can see from the graph, the incidence of bleeding occurs in multiple locations, but primarily in the joints. There's also impacts in soft tissue and muscle. But the joint bleeding, known as hemarthrosis, is typically in the ankles and knees, is accompanied by pain and swelling, and really can result in disability. So Altuvio, also known as efanesoctocog alpha, was approved in February of 2023 for adults and children for routine prophylaxis, on-demand treatment and control of bleeding episodes, and perioperative management of bleeding. It was specifically designed to maintain high sustained factor VIII activity levels and prolonged half-life. It can be dosed once weekly, which was a substantial improvement over the other available therapies that were available at the time. And it had significant benefits that were recognized by multiple health authorities. As you can see, just from the United States alone, the FDA designated it a fast track application. It received breakthrough therapy designation, as well as a priority review. It was also recognized by multiple other health authorities worldwide as an advancement. The study that we'll be discussing is the primary study for approval. It was an open label phase 3 study of Altuvio in patients that were previously treated with factor VIII recombinant products. It was called the Extend-1 study. And it included adult patients that were greater than 12 years of age and adolescents as well that had hemophilia A. And they had to be on previous treatment with other recombinant or plasma-derived products. It was a novel approach in that we had a pre-study where we allowed all comers to enter into the program. And from there, we were able to move them into different arms, whether it's a prophylaxis arm or the on-demand treatment arm. The primary endpoint was the comparison within arm A. That was the annual bleed rate in the prophylaxis treatment arm. We also had an important key secondary endpoint that was negotiated with health authorities. That was an intrapatient annual bleed rate comparison that was utilized and also was requested by certain health authorities, particularly the EMA. And we had extensive discussions with health authorities, including the FDA, on this particular endpoint. It was a novel approach, but one that was necessary, and we'll go into more details on the secondary endpoint in the next slide. There were also many other secondary endpoints: patient-reported outcomes, quality of life. We had understanding of the pharmacokinetics, many safety, as well as understanding of the treatment bleed. So in terms of the key secondary endpoint, it was a comparison of the pre-study population along with the arm A, which was the interventional component of the study. The analysis included a subset of patients that participated in both arms and had to have at least 6 months of available therapy. We had a non-inferiority analysis followed by a superiority analysis, and that information is displayed in the graph on the right, where you can show that the estimated mean ABR reduction was 77%, which was demonstrating superiority and had a statistically meaningful p-value. So addressing challenges in rare disease is really important. It's not, as many panelists had noted, it's not something that's common that you can actually have a randomized controlled study. It faces multiple limitations, small patient populations, ethical concerns, particularly around including the placebo patients or suboptimal treatment arms, as well as in hemophilia particularly, you had high interpatient variability in the bleeding phenotype as well as the treatment response, which made it difficult to compare. So we had long discussions to have an interpatient comparison, and ultimately what we saw is that it overall enhanced sensitivity and reduce variability in the comparison of the pre-study population compared to the interventional period. We felt that this design controls for the interpatient variability and baseline bleeding risk, enhances statistical power despite the modest sample size, and provides clinically meaningful benchmark for assessing treatment benefit. In terms of the regulatory history, it's important to note that a paradigm shift had changed at the time. All other agents were approved comparing prophylaxis versus on-demand treatment, but the paradigm shift was that most patients were only on prophylactic treatment. So making that comparison was unethical. You couldn't put patients that were on prophylactic treatment on an on-demand arm, which made it essential to think about another alternative approach. There was long discussions about using intrapatient comparison as the primary endpoint. However, there were also risks and concerns in using that as the primary endpoint, ones that we were not sure would come to fruition at the time. We did get agreement on having an annual bleed rate as the primary endpoint, and that was based on an estimate approach. And ultimately, we did see a positive impact on the annual bleed rate as the primary endpoint, as well as the interpatient comparison. And as I noted in the previous slide, we really do believe that this interpatient variability, that its interpatient comparison really enhanced the sensitivity and overall reduced variability amongst the comparison. And we were very fortunate that we had thought ahead and had this baseline study, the observational pre-study, that allowed important data integrity elements to be captured without bias, and also for us provided that flexibility so that we can actually include patients in that pre-study into different arms, treatment arms. In terms of future considerations, it's really important to think about interpatient comparisons and thinking that they can be leveraged, particularly for these small patient populations, in order to support regulatory decision-making. I think we talked a lot about the early interactions and frequent interactions that need to happen. And that was essential in this particular instance too. More frequent interaction with health authorities really can avoid missteps, particularly early on as you really try to design these studies and figure out how to leverage the observational data in order to support approval as well as labeling. We did end up with a statement in the label on this arm. However, it did not have the p-value. And we actually had limited secondary endpoints that were included based on the statistical methods that were chosen. So it is very important to have those discussions early on, not only on the design, but also what the ultimate labeling discussions would look like. It's also important to know that consistency will be important, not just for FDA, but across all health authorities, because EMA wanted different endpoints, as well as FDA had their own ideas, but we're talking about a very small patient population. It's really important to have a very consistent approach to leverage the data that we have in order to support the ultimate results and labeling that we can have in those discussions in advance. We also noticed differences even between different divisions and the Center of Drug Evaluation Research, as well as Center of Biologics Evaluation and Research. It's really important to, and it was discussed many times during the panel, as well as in multiple different forums, that to have this consistent approach across health authorities and across single agencies. And with that, I believe this ends my portion of the presentation, and we can move to the panel discussion. Oops.
Thanks so much, Todd. That was really helpful to ground us in our conversation now. At this point, I'd like to invite the entire panel up, and I will introduce each of them. A lot of outdoor noise here in DC. We'll give it 1 minute. Okay, great. Again, really excited about this incredible panel we have gathered here today. We've introduced Todd. I'm going to sort of go down the line here. First, introducing Jen McNary next to Todd. Jen is a trusted voice in the rare disease community as a mother, public speaker, and fierce advocate. She's the co-founder and principal of Canary Advisors, which is a boutique patient advocacy consulting firm with a focus on regulatory and access patient engagement. Next, we have Dr. Alison Berendt, who is the Chief Science Officer at the Foundation of Angelman Syndrome Therapeutics, or FAST. Next, we have Dr. Adora Nadeau, who is the Chief Regulatory Officer and Executive Vice President of Portfolio Strategy and Management at BridgeBio Pharma, where she oversees regulatory, quality, government affairs, and program management. Next, we have Dr. Rebecca Rothwell-Chu, who is a supervisory mathematical statistician in the Division of Biometrics 4 in the Office of Biostatistics at the Center for Drug Evaluation and Research, or CDER, at FDA. FDA is famous for having very long titles, so we know that. Finally, I'd like to introduce Dr. Tingjing Zhou, who is lead mathematical statistician in the Office of Biostatistics and Pharmacovigilance at the Center for Biologics and Evaluation and Research, or CBER, at FDA. So what we're going to do is ask each of our panelists to provide some introductory remarks, telling us a little bit about themselves and their organization. And then we'll get into some moderated questions. I do want to remind everybody that throughout, you can feel free to submit questions through the Zoom Q&A function for those who are online. And right now we'll start with Jen going down the line.
All right, that was a surprise. Okay, great. So thank you very much first to the organizers for the invitation to contribute. I'm really pleased to be joining some fantastic representatives of the patient community that you've already heard from and that you will hear from later today. I wear a lot of hats, but today, most importantly, I'm the mother to 3 amazing guys and a few girls and extra kids here and there too, to those that know me. My 2 oldest sons were diagnosed with Duchenne in February of 2002. My oldest son, Austin, died in February of 2025. He was 26. It was not enough time, but because he had access to an early-ish diagnosis and the opportunity to participate in 2 open label studies, one for exon skipping and one for gene therapy, he outlived and outperformed his life expectancy. He had a chance to live as an adult, and that's not lost on me. He made a huge impact on not only the people around him, but the entire rare disease community via his participation in drug development and, most importantly, regulatory advocacy. His brother Max is 23 and has had a similar impact on the world. He's been participating in clinical trials since he was 9 years old. And my 3rd son, James, is newly 18 and away at college. And he's managing his own dual diagnosis of common variable immune deficiency and congenital cholesteatoma. So this issue of rare disease clinical trials is near and dear to our family's heart. In addition to raising my biological, adopted, and foster children, I am the co-founder of One Rare and Canary Advisors, as mentioned. I'm focused wholly on different aspects involved in those living with rare conditions. I share in such detail about my sons because I want to convey today that participation in clinical trials is not just a time point or an event in a patient's life. If we're lucky, it's something that they have to weave into the entirety of their lifetime, like Austin did. He died in the midst of a clinical trial, and he's been participating in them since he was a toddler. It's healthcare. Clinical research for small, rare, and ultra-rare populations is often the only opportunity for a treatment. When you realize that families are being asked to incorporate often multiple clinical trials at the same time, and often with multiple siblings and other family members into their work, school, and family lives, we know that we need to reduce burdens and increase accessibility to all. We have to design a study for maximum interpretability and for the best outcomes possible. It's such a miss if we're not collecting endpoints that matter to the clients, the patients, in a way that's meaningful to regulators. And it is the worst case scenario when the benefits of a product are apparent to the patient community and to the clinicians and caregivers, but not showing up in an approvable way in the clinical research setting to the regulators. In addition to personal experience as a caregiver, to patients in clinical trials, and through my work at Canary Advisors, I've learned a lot about what matters in the design of clinical trials to patients. For example, only utilizing a placebo for small time periods and when absolutely necessary due to the absence of a well-understood natural history or registries. I spend a lot of my time focused on ELP-FDD meetings or patient experience data collection during an interventional study. This is a great way to be sure that we understand the benefits to a patient population where it's difficult to power a study. In addition, in diseases like Duchenne, we have moved away from individual treatment options. And the discussion in our patient community, happily, is now about combination therapies. The patients understand the benefit of coming out of disease from many different angles, and all of these are important. And they appreciate the additional noticeable benefits of stacking therapies. The real challenge there has been in the payer world, but we're poised to handle that. In the same way that we need to trial combined therapies, there also needs to be continued focus on speeding up the development by taking what we know about one therapy and applying it to the next, especially where it has the same mechanism of action, instead of treating each new therapy as a new entity. There needs to be a movement towards a new gold standard of rare drug development that pushes past the set of standards in place for common conditions and acknowledges the challenges of working in small, heterogeneous populations with difficult and often non-validated endpoints. Placebos are not only undesirable, as mentioned in the first panel, but can actually be unreliable in small data sets, which can cause studies to miss endpoints despite efficacy. Lastly, whatever trial design is ultimately decided on between the company sponsor and the FDA, and if we're lucky, the patients, what patient communities need most is a predictability, consistency, flexibility, and urgency at the regulatory level. Because what was mentioned in the beginning of today, and I'm going to reiterate, is that time is muscle, vision, function, whatever your disease, and that it is incredibly important that we stick to whatever we've decided on and do the best we can for patients. Thanks.
Great. Thanks, Allison.
Well, I'd like to thank Duke-Margolis and the FDA for giving me the invitation to be here and speak to you today about my lived experience. I also sit here wearing many hats, quite different and also quite similar to Jen. I'm uniquely positioned in that I'm a scientist, I'm a clinician, I am a sponsor, I am part of industry, and I am most importantly the parent of a beautiful little girl who lives with a very debilitating disorder called Angelman syndrome. I am a veterinarian by training. I'm an interventional radiologist and an internist, and I've been working in clinical trials developing medical devices for naturally occurring diseases for my whole career. And when my daughter was diagnosed with Angelman syndrome at 5 months of age, I was naive enough to believe it was easier to fix a single gene disorder than a feline ureter. I was wrong. We've solved the feline ureter problem, but we have not yet solved the single gene disorder problem. But with events like this, I think we're well on our way. And I jumped in head first. I joined the Foundation for Angelman Syndrome Therapeutics, which was the only organization that was singularly focused on developing meaningful and transformative treatments for this disorder. Industry was not interested in such a rare disease in 2015. There was incredible work that had been done on the backs of amazing scientists in academia. And there was a huge valley of death that was very clearly before us in that there was drugs being developed for mice, and there were clinicians that wanted to treat my daughter. And there was absolutely no bridge that was being formed between the academic and industry. And we unfortunately had to start our own company in order to advance a therapy for humans because industry wasn't doing it and we couldn't wait any longer. And so our foundation founded the first disease-targeted therapy, an antisense oligonucleotide. We started a company called Genetics Biotherapeutics, and we got from proof of concept to first in human. We ultimately partnered that program with a company called Ultragenyx Pharmaceuticals, who acquired the company, and they actually just completed enrollment of their phase 3 with a sham-controlled study in 7 months of 120 patients, which is very clearly that our community is ready and our community is able and incredibly educated. Other companies, 2 additional companies are developing ASOs, both with placebo-controlled studies, and those are enrolling rapidly as well. With that, I actually serve also as the Chief Development Officer of an accelerator called AS2 Bio, where we are accelerating gene and cell therapy for Angelman syndrome, a hematopoietic stem cell gene therapy, and an AAV gene therapy. And we cleared our IND for our AAV gene therapy just in April, and we'll be dosing our first patient in the coming months. But what I can say is a really important job I also have is that I am the co-director of the Angelman Syndrome Biomarker and Outcome Measure Consortium, which is a pre-competitive consortium that supports 16 pharmaceutical companies, as well as patient advocacy groups, academics, and clinicians, in order to pre-competitively develop endpoints and biomarkers that are meaningful and measurable. And what I can tell you is the only people that understand what's meaningful are people like us sitting on this stage. And it's very hard for others to try to tell us what is meaningful. You can tell us certainly what's measurable. And so what I can say is, as a patient advocate and as somebody who really understands patient-focused drug development, I really have to think about what we know in controls. What we know is that we have a 20-year natural history study of 600 patients in Angelman syndrome prospectively that has been morphed and designed to model clinical trials that are currently ongoing right now. And essentially, the Biomarker and Outcome Measure Consortium has essentially developed a master protocol working very hard to ensure that every trial uses the same endpoints and biomarkers so that we can borrow from each other with the ultimate goal that that is a responsibility of industry to share controlled data in order for other sponsors coming up and behind and the community to not have to continue to enroll in controlled studies. What I can tell you is, as somebody who has run focus groups in understanding willingness to participate in gene and cell therapy clinical trials, there's huge interest. but not if there is a controlled study in which they would have to be immunosuppressed or receive chemotherapy in order to not know who is in the controlled trial or not. That is not an option for our community, and it shouldn't be an option for rare disease communities. So as a patient voice who really has to wear the hat of what is meaningful, what is measurable, and how can we understand the willingness to participate, and how do we ensure patient voice is heard, I could tell you that benefit risk tolerance should really only be determined by those that are affected by the disease. Nobody can understand the risk of doing nothing until you spend a single day in the life of a family who has a child that really needs a treatment desperately. And just because a disorder is not degenerative does not mean it's not horribly debilitating. So when we talk about being tolerant and being flexible, we have to think about debilitating disorders, rare disorders, and not just those that are degenerative, because living with a disease for your entire life without a single semblance of independence is sometimes nearly as bad. So we really have to talk to families at every turn to understand what they need, what they want, and what they hope for. Because oftentimes the risk of doing nothing completely outweighs the risk of doing something with a semblance of hope. Parents are smart and they understand we need to bring them into the conversation as clinicians, as scientists, and as sponsors at every turn. That is patient-focused drug development.
Well said. You. I think that's a good transition. Just really briefly, Adore and Do, as Rachel mentioned, I'm the Chief Regulatory Affairs Officer with Bridge Biopharma. I've been here for almost 4 years. I've spent most of my career in regulatory drug development policy, spent almost 10 years at FDA. So certainly understand some of the challenges that the agency faces with some of these decisions with regards to the choice of controls across, especially rare diseases where it is especially relevant. What I would say is that Bridge Bio, one of the reasons I joined Bridge Bio is one, not just our focus on science, but also on bringing in the patient voice. And I know that's been a key theme across at least the earlier panel and will be for this panel as well. And so, you know, we've done every decision that we make is founded and grounded on what's the best for the patients, for the patient community. And that's one of the reasons I joined. We're a young company. We were founded in 2015. We received our first 2 approvals in 2021, both in ultra-rare diseases. The first was for a disease known as MoCD type A that at the time impacted only about 400 patients globally. And we leveraged an external controls, very close collaboration with the FDA to get that treatment approved. The second approval was for another rare disease, a rare form of cholangiocarcinoma, also approved in 2021. We received our third approval in November of 2024 for Attruby, a treatment for ATTR cardiomyopathy, a larger rare, but rare disease nonetheless. And we have other products that are currently in our pipeline, later stage. We're doing work. We're developing a small molecule for limb-girdle muscular dystrophy type 2I. There are over 30 subtypes of limb-girdle muscular dystrophy and not a single treatment approved. So we're hopeful that we will be able to bring treatments to patients. We're also developing a treatment for ADH1, autosomal dominant hypocalcemia type 1, which is another rare disease. We're developing a small molecule there. And there we've actually leveraged an internal control, which is relevant for our discussion here. That condition presents from an imbalance of urinary and serum calcium. And so our treatment, which is calcilytic, is an antagonist to the gene where we see the gain of function mutation. And so, you know, when you're looking at some of the our goal of establishing mineral homeostasis, it presents a unique opportunity to leverage an internal control, looking at patients at baseline, following them after treatment, and assessing the treatment outcomes in that setting. But we're a company that's focused on addressing some of the most difficult genetic diseases. And, you know, it's inherent in how we were founded. It's inherent in our science. It's inherent in the diseases that we pursue, including those that we refuse to give up on because we believe in the science and the work that we do with the communities that we serve. And so I'm really excited to be on this panel. Thank you to Duke-Margolis and to FDA for inviting us to join the first RISE panel. Hopefully it's the first of many where we get to discuss and address some of the really challenging issues and questions that we face in trying to address some of the diseases with the highest unmet needs. And that's why BridgeBio was founded. That's why we exist. And we hope to continue to partner with all the relevant stakeholders in this space. Thank you.
Thanks, Victoria. Dr. Chu.
Thank you. Hi, everyone. I first want to also acknowledge the organizers for putting this outstanding meeting together and already learning a lot today just from the first session and the beginning of this discussion. Just to briefly introduce myself, my name is Becky Chu. You heard all the acronyms already, but I am a statistician in the Center for Drug Evaluation and Research, and specifically, I supervise the team that provides statistical support for the Division of Rare Diseases and Medical Genetics. So this division regulates drugs and biologics that are intended to prevent or treat rare inborn errors of metabolism. So as I'm sure you can imagine, we often are faced with helping to design and evaluate trials in really small, really small sometimes patient populations. And I know you all are very familiar, but just to touch on a couple of those challenges that we face that often lead us to end up with kind of inconclusive results at the end of a trial. Some of those challenges include some poorly characterized natural history due to limited patient populations, some lack of consensus on endpoints, and again, that's often due to the limited ability to perform those early phase trials that we can do in the common disease setting. We touched on earlier in the last panel, the heterogeneous and highly variable disease presentations, and in some cases, the slow disease progression that results in some untenably long trials. So it just, some of those challenges are evident outside of the rare disease space, but I think the limited number of patients that we have to address those challenges really determines what is tenable in this rare disease space. So I think today, as we go through the questions, I'm hoping to kind of point to what different aspects may influence our choice of the design, and in particular, as the topic today is the choice of the control. I think I've come to acknowledge that most rare disease programs are highly unique and do need a case-by-case evaluation. I was interested in the archetype discussion earlier, but I do think there is nuance to a lot of the different rare disease contexts and how this might impact some of the acceptability of some of the flexibilities that we consider. So I hope that we'll continue to discuss that in this session and think about some of the creativity that might be applied in some cases versus others.
Thank you. Dr. Zopp.
Okay. Hi, everybody. Thank you for having me, and thank you for the organizer for putting this panel together. My name is Ting Ting, and I'm a statistical team leader at the Center for Biologic Evaluation and Research, so CBER FDA. So my team and I, we provide regulatory statistical support to the Office of Therapeutic Products, OTP, at CBER. We review mostly gene and cell therapy trials in different therapeutic areas, such as neurology, ophthalmology, and cardiology and nephrology. So some of the four major therapeutic areas that we review in. So I do see a broad range of applications in different disease areas. When it comes to rare disease, we do recognize the unique challenges those trials face, because there's a small patient population, recruitment might be difficult. And sometimes with rare disease, there's limited external information, which make it challenging to even select a meaningful clinical endpoint. So to help address some of those challenges. So FDA, there's a collaboration between CBER and CDER. We have recently launched this rare disease endpoint advancement program, pilot program. So the initiative is to support the development of novel efficacy endpoints for therapies targeting rare diseases. And before, we talk about the different type of controls that can be used. I just wanted to kind of touch upon like the definition of rare disease. So the rare disease is often defined as a condition that affects less than 200,000 people in the US. So, there's a wide range of what constitutes as rare disease. And we do, kind of distinguish between like the more common rare diseases and the really truly rare one, the ultra rare ones, right? For the more common rare diseases, I think, randomized concurrent control trials are usually feasible, and they might be preferred because they tend to generate the most robust and interpretable data. But for the really rare one, ultra rare ones, we recognize there is a greater need for regulatory flexibility in trial design. So in terms of selecting like an appropriate internal control, in my experience, I think it's very program specific. It depends on the disease, the therapy, the endpoint, and the available data, and what's practical for the specific development program. And so there's some unique challenges in gene and cell therapy trials. For example, with gene and cell therapy, it's a one-time treatment, and they are meant to be long-term. So some traditional like crossover designs where you might compare within patient for some efficiency to improve efficiency, those kind of designs are not really are not feasible in gene and cell therapy trials. And some randomized or even active control trials, we're actually comparing to an approved therapy or gene therapy are also very rare in gene and cell therapies because of practical reason and cost constraints. And we'll talk more about those internal controls. And I look forward to the discussion and hear everybody's perspective and insights. Thank you.
Thank you, Tintin. So we have a series of questions. As you can tell, this panel has an incredible diversity of perspectives represented. And so I want to walk through some questions that we give each of our panelists an opportunity to respond to. Starting out at sort of a higher level around just the role of internal controls in these clinical trials for small and diminishing patient populations. And Todd, maybe we can start with you to speak to not only the role of this model of internal controls, but also some of the challenges and ethical considerations that you have to go through as a sponsor when deciding on the nature and type of that internal control.
Yeah, thank you. So of course, the case study was a good example of leveraging a population that's already treated. And certainly what we saw with the paradigm shift, moving from an ability to compare to different types of treatment, whether it's on demand or prophylaxis, that was really essential in the design to allow for an alternate approach. And it took a good amount of statistical rigor and understanding, and essentially what we saw was it really enhanced sensitivity, reduced all variability. The patients, when you had an interpatient analysis, there because of the differences in treatment response to the bleeding response, the phenotype, the analysis was certainly well suited to leverage the interpatient comparison. That's not always the case, and we were fortunate enough to have this pre-study. We were fortunate enough to have historical controls. We were fortunate enough to have an area where treatments had been available for many years. When you get to the rare and ultra rare, it's a much more difficult approach. There's ethical standards. We talked about not being able to do randomized controlled trials, not being able to have placebo arms, really trying to understand the population and do a more of a comparison, whether it's historical or one that you would have collected data for a certain period of time as these therapies are being developed. So certainly there's different options, but ultimately it's very important to design it based on the disease state, but also the population in question.
Great. Thanks for that. Jen, Allison, anyone else want to weigh in on that?
I'm going to make it really personal. I mentioned that my older son passed away in February and that his younger brother is still alive. The age difference between the two of them is 3 years. There's also approximately 3 years between the time that Max started treatment on exon skipping and the time that Austin got access to exon skipping because of the trial design. It was intended to exclude those that had stopped walking. I mention this because there are things that you cannot get back. When you have a degenerative disease, there is time, there is muscle, there's cardiac function that you cannot get back. And so I actually am challenged to think of a scenario where it's okay to withhold a potential therapeutic from a group of patients who, could, progress in their disease without access to that treatment. So I also know that there are certain times and certain scenarios where a very short time of an alternative, a standard of care, or with a quick crossover, there might be scenarios where that's possible. But I would challenge folks to think of that the exception rather than the rule, right? Let's start by thinking, how can we not utilize a placebo comparator, and then move that way rather than starting from, gold standard is placebo comparator and then trying to move in the other direction. And I also want to point out that there are a lot of opportunities to get all your questions answered that don't include forcing decline in a patient population, right? So real world evidence is a really great opportunity for that. We can follow patients like my family are willing to literally do clinical trials. Austin had just had a clinical trial visit in January. He died in February. That's challenging to get patients and end-stage diseases to clinics, but we do it. And so there are tons of opportunities to continue to follow these patients and get all the answers to all of the scientific questions that we want and to become sure decades down the line. But if we're worried about this generation of patients, the answer is never placebo. And I realize that my opinion may differ from those in the room, but that's why I'm here.
I don't think my opinion is so different. I think I come at it from a little bit of a different perspective in that I think it's our obligation as an industry and patient advocacy and the regulatory authorities to not only encourage, but actually require companies to share placebo or controlled data. Because what happens is that everyone holds this data to themselves. It's expensive data, it's valuable data, and we all understand that. But each patient is not a data point. Each patient is a life, is a family, is a sacrifice. It's not only money to the company. Imagine the money of that family that they have spent missing work, missing school, missing opportunities. And if we can share that data, and there are beautiful examples of multiple sclerosis and larger indications in which that has been done post-approval. But in rare disease, we don't have that luxury. Angelman's unique in that we have 9 different modalities that are being advanced for Angelman syndrome, and our foundation is advancing most of them. So we will share our data with ourselves. But what we don't have are that the controlled studies are on the CEDAR side, the ASO side, in which there's, it's redosing, it's not a gene therapy. There should be an obligation, in my mind, for these members of industry and the community to share that data to the community, de-identified, not company specific, so it can be borrowed. Because then you truly have scientific rigor. And we understand scientific rigor has to matter, but when we look at statistics, we can't look at these patients as a data point in which it's a big enough indication that it should require a randomized controlled study. That's not possible for these communities. And so how do we do better? It's the requirements for approvals that you must share your data or you won't have an approved drug. Then guess what? Everyone's going to share their data. So you have to, it has to come from those that matter to industry, which is the regulatory authorities, in my opinion, rather than the community begging companies to share data, of which they often don't do. I think that's the path to really be able to change the vision where there is already controlled information. And very ultra rare, you're never going to have that controlled information. Run-ins are where you're going to have to start.
Great.
Yeah, I mean, I would agree with a lot of what's been said. I mean, there are certainly a lot of challenges as well as opportunities. And what I'm hearing from, what my fellow panelists have shared is the importance of starting with hearing from the patient community, especially as we're designing the protocols. And then also bringing that voice into the regulatory discussions with the FDA to align on what is the ideal trial design for that particular disease, as they are all different. And so when we start to think about, rare diseases and what we all know, which is the disease heterogeneity, et cetera, and which of these diseases would be appropriately structured with an internal control within the context of a clinical trial. It could vary, right? What do we know about the natural history of the disease? What do we know about the disease progression? How heterogeneous is it from patient to patient? Is this reversible? Are there biomarkers that can be easily measured? Are the biomarkers relatively stable over time? Is this a condition where we can have a run-in period where we're evaluating some biochemical measures and, introduce the intervention and then compare and evaluate? And so this could look different from disease to disease. I do think, though, that there's an opportunity to really enable those discussions with baseline controls with the agencies and, talking through and aligning on when is it appropriate to leverage a baseline control and really those principles that would enable more efficient use of baseline controls. Now, with randomized withdrawals, I know the challenge there is the period where you are withdrawing a therapy that potentially is providing benefit. And, it introduces some of the same ethical considerations that we encounter with placebo-controlled studies. So I do think also looking at the disease, understanding the patient perspective, understanding, is this a disease that is progressive? Is this a disease that is irreversible? Is it, time is muscle? What exactly, how are the patients experiencing it? What is the burden? And sort of aligning on whether treatment trial approaches such as randomized withdrawal or delayed starts, some of these other opportunities are appropriate. But I do think that when we look at these internal control options compared to the gold standard of placebo-controlled, sometimes very extended periods of placebo, I think arguably being able to leverage some of these alternative designs is probably ideal, both from patient perspective, drug development perspective, and really efficiency in being able to get these treatments to patients as quickly as possible.
Great. Thank you, Adora. I think it would be great to hear sort of the FDA perspective on this question around what are the opportunities and then also the challenges from your perspectives around the use of internal controls.
Sure, I can start. I won't claim to provide the entire FDA perspective, but I'll certainly give my perspective. Yeah, so I think when we're discussing internal controls, and I think we've already gotten there a little bit, that there's obviously a distinction in this bucket of internal controls of non-randomized controls, which includes like baseline controls and randomized controls. And I think those baseline controls, we can think a little more similarly like we do about external controls, which I know we'll talk about in the next session as well. And I do think there is a place for them, but we need to consider the circumstance that a baseline control might be appropriate. What we like about randomization is that it removes the systematic differences between treatment arms in both known and unknown prognostic factors. So that means that when we see a difference between the arms, we know that it can be attributed to the treatment itself. That said, there are places where a randomized control arm wouldn't be necessary in order to still provide us with interpretable results. And that's really when we have a good understanding of the disease, usually with a reliable natural history, that patients wouldn't have some kind of spontaneous improvement, if improvement is the goal, on a well-defined and a meaningful endpoint for patients. And also when the drug is really expected to have a large effect. This helps us kind of battle against the bias that we often see in the non-randomized controlled setting. Unfortunately, I do acknowledge that in the case of these small patient populations, we often don't have that strong natural history of the disease. And that does mean that randomized controlled trials are sometimes the better way to obtain interpretable results. I will say, I think in rare diseases and in clinical trials in general, we're often faced with balancing a trade-off between precision and power with bias and accuracy. And I think that it may be preferable to make compromises in the precision space or the increased variability space. And that might be in terms of like flexibility of a pre-specified statistical threshold, doing something different than that 0.05. I think I would rather compromise in that variability side rather than on compromises in terms of accuracy or bias, which unfortunately may arise in the kind of external control or non-randomized control setting. Those are just a couple of thoughts. The last thing I'll mention, I think we heard today earlier by our leadership, and some of those things were new to me to hear as well, and very exciting new pathways coming. I think there's a lot of emphasis on other information that can support what we call the adequate and well-controlled trial. Often the strength of that other information can counter some of the weaknesses in our adequate and well-controlled trial. So we're still looking for that trial, but just to say that that supportive information is really important as well. And I think we try to incorporate some of the points that have been made already and coming into those packages. I'll stop there.
Yeah, hi. So a lot of what Becky has said also applies to gene and cell therapies. So we do recognize the challenges of running trials with for rare diseases. Randomized placebo control. It is the gold standard because it's randomization kind of removed all the measure confounding. Sorry, this randomization helps balance the known and unknown confoundings across treatment arms. But that being said, we do not, what kind of internal control is appropriate is really program specific. We never ask a sponsor to only consider randomized placebo control trial. The type of control, it really depends on the disease, the endpoint, the therapy, and what available data we have already, and what is practical for the program. So in terms of the baseline control, there's been some regulatory precedent with gene and cell therapies. I'm aware of, there's been a couple therapies that have been approved based on pre and post treatment. And sorry. So with the baseline control, right, you are actually comparing pre and post treatment. And so you're actually using each patient as their own control. It improves efficiency. So sorry. So I think with the baseline control, so the benefit is everybody. I think it helps with recruitment because all the patients get treated. But I guess the limitation with baseline control is sometimes it's hard to separate the true treatment effect from the placebo response because there's a lack of blinding in the natural fluctuations and variability in a disease or even regression to the mean. So if you have a really slowly progressive variable diseases, there's a lot of uncertainty. So that's a limitation. But I think if you see there's a large treatment effect observed, it helps us make the conclusion that there is efficacy. Yeah, that's
Great, really useful insights. Thank you both. I want to return to a sort of theme that came up a lot during the first session, which is sort of where and when and how to incorporate the patient voice in the process, and particularly in this instance around when and how do we use internal controls. And so for Jen and Allison, how can we improve upon the use of internal controls so that they're more beneficial for patients? And how can patients have a stronger voice in sort of when and how to use these?
You want to start? Sure. It's a great question, and I appreciate it actually very much. I think the patient voice, and in my case, and in the case of neurodevelopmental disorders, it's the parent and caregiver voice, because our children don't speak a single word, which is part of the challenge, is that they should be present and have a voice at every stage. And I think certainly as it relates to sponsors, as it relates to study design, as it relates to endpoint choice, meaningfulness, what is meaningful, what's measurable, what's tolerated, and I think almost most importantly, what is the risk-benefit tolerance to that community and to that disease itself. But as we think about internal controls and what would matter, I think what's really important to remember, particularly in rare disease communities, is that even though the disorder may be nearly identical, the heterogeneity is profound. And when you have a disorder that doesn't affect one symptom, but affects a gamut of symptoms, from cognition to communication to motor to fine motor to seizures, to essentially every part of daily living, certain patients are more responsive than other patients to different domains. And that's where I think having that baseline understanding of that particular patient, of what you don't know at the beginning of a trial, if that patient's going to improve in motor, in communication, in cognition, or in nothing. And that's where natural history data, in my opinion, is really important. But the problem is, and to the panel before, it gets drowned out in noise when there's so much variability. So every patient is nonverbal, right? So therefore, if there's a change in receptive or expressive language, that's a change. You can't placebo that. You can't make my kids speak. I promise you, if you could, I'd give her that sugar pill every day. But if they start speaking, of course, that's a huge treatment effect. But you may not get that, right? But you may get a kid that starts using their iPad, that starts signing, that is focusing, that's listening, that's following instructions, that can coordinate their body and swim and not drown, which is the leading cause of death in Angelman syndrome, because it's not degenerative, but they're obsessed with water, so they can drown. These are the things that one patient may learn to swim, one patient may learn to walk, one patient may learn to run. Those are all huge changes. What's the average? Walking. If the kid already could walk, that kid had no effect. If the kid couldn't walk, that kid had an effect. The median is no effect. So you have to look at the individuals. And that's where I think when we think about composites, which everyone hates, at the FDA particularly, families love it, by the way, because it captures their children. It really could capture their children in a way in which you can figure out where they're responders or where they're not, and truly try to understand the treatment effect. So for us, being your own control in a composite can paint a beautiful heat map of what's actually happening to that individual that you may not get with averages and statistics based on the average member of the community, if that makes sense.
Definitely. Great.
I'm glad I let you go first. You know, I can't agree more really, because when patients and caregivers are looking at improvements or benefits, they're looking at their own child, right? And so oftentimes it's what is that child? What was the biggest pain point for that child or that person prior to the study, and then what changed over time? And what changed may have been that they didn't get worse, and that's really hard to measure in a clinical trial, and it's also really hard to measure against the progress of another patient, right? Because every child's or person's trajectory in a disease is different. And so another vote for, figuring out ways, people smarter than I am, to design studies that look at that person, that human being, and how their changes over time. And I'll be personal about it today, again, because what do I know? I know my children. And I know that Max, when he was diagnosed, was supposed to follow one path. And Max today is 23, getting ready for a trip to Sweden to play power hockey, go Volts, go Boston Whiplash. I think it's really important to think about what can a person accomplish over time with small changes in their path. He still looks like a very disabled human being, but I consider the trials that he's been part of to be a success. And so I think that's really important. What does success look like to a statistician versus to a mom is very different. And it's worth noting that there have been significant arguments over time for both of these products that my son has had success with. And I am very grateful that we managed to get access to other people through these various channels. I think it's also really important, if we're talking about how do we start to think about including the patients and including perspectives over time, it's from the very inception. We can answer questions like Allison mentioned, will there be a placebo effect? I promise that if will and want would have saved anybody's life, it would have been Austin. And those of you who know Austin know that there is no person that wanted more to benefit from life. He lived a really hard life, and he wanted to walk, and he stopped walking at 10. And he wanted to live, and he stopped living at 26. And so you cannot do that in Duchenne. And I would venture to say in most genetic diseases, we cannot, that sort of placebo effect is going to end eventually. And so real world evidence picks up where clinical trials end. And hopefully you're making the mistake of giving access to a broader community and then finding out it wasn't as effective as you wanted it to be, or it could have been, rather than the other, which is allowing a whole population of patients to decline where you could have been preventing that decline, right? So you don't want to make that type 2 error. As the late, the former commissioner, Janet Woodcock would say, you want to err on the side of being patient-centric and learning that it wasn't as good as you thought it was later on. So I think those are really my thoughts. And again, we all want things to be effective. We all realize that not every drug that starts is going to finish. We know that the failure rate is huge, as we heard in the previous panel, but failing in that way of taking things all the way to the finish line and failing in the real world is a lot better than kind of leaving something on the shelf when it could have been something great in the community.
Maybe I'll add to that. I mean, I agree with what you both have shared. I do think, the concept of a placebo effect is very real. That from a drug development perspective, we are very conscious of. What I would say that's why the natural history becomes really important, understanding what the natural progression should be and understanding what outcomes would not be present but for an intervention. And I think that is an area, there are many areas where the patient voice is critical, but that's an area where hearing from communities is so critical so that we can contextualize the data that we are seeing and we can contextualize the impact. There are 3 themes that have come up, during this panel today and is also quite present in FDA's guidances, and that includes the idea of the endpoints being well-defined, clinically meaningful, and there being a large effect. These 3 points are at the core problematic in rare diseases because quite often we're dealing in spaces where there is no precedent. We don't know what's well-defined. We're still trying to understand the clinical meaningfulness of these outcomes. And then the question of how large is the effect and is it large enough? That's also something that we continue to have to understand and define. And so I think being able to contextualize this from disease to disease in those interactions from a regulatory perspective becomes very important. Otherwise, we leave a lot of products at the table that really should be in the hands of patients because we don't understand whether it's clinically meaningful. But if we talk to patients and families and caregivers, they will tell us this is what my child is now doing. And without this treatment, they wouldn't be walking. They wouldn't be, having their eye movements. They can see me, they can smile, and those things are meaningful to families. To your question, Rachel, about when to bring patient voice in, I would say early and often. I know people hate that statement. I mean, it is true. It's the reality. But I think being able to further define that really helps us mobilize that and enable that in our drug development efforts. I'll give you one example of where it was critical. And this was in my prior life, development of a treatment for PKU in adults. And in this situation, the challenge there was, this was a randomized withdrawal. There was a question about whether, the outcomes were clinically meaningful. Given that improvements in cognition we're not seeing in the context of the trial, but we understand PKU. We understand that reductions in blood Phe, should lead to some certain outcomes based on the history of the disease and what we know. But yet this was sort of a back and forth discussion around whether the reductions in blood Phe were clinically meaningful, whether they were important to patients. And so we actually had a joint meeting with the agency, with the patient groups and patients that were in the trials, as well as PIs, and so impactful hearing from each of these participants what it meant for them to have control of their blood phenylalanine. And I think what that did was it allowed the agency to contextualize the data, the improvements that they were seeing in the absence of a statistically significant improvement in cognition. This is what it means to each individual patient that is receiving this treatment and how it has changed their lives and the impact it has on their overall trajectory, their activities of daily living, et cetera. And so, especially in rare diseases where, again, very unprecedented in many cases where we are all learning together, right? We're learning from patients, we're learning from developers and academics, we're learning from the agency's learning from us. I think the ability to walk into these programs really collaboratively to understand, what are some of the nuances with this particular disease that would prevent it from fitting in this particular box, or would establish the premise of the increased importance of the patient voice so that we know what it means when a child can move their eyes to follow their parents from one side of the room to another. That's important to some families, but when you look at it in a review document and you see that, 2 patients had, X improvement, it may not mean much from a review perspective. And so really being able to bring in families, patients, developers together to really understand what it means, I think is so critical.
Right. And I would also just add, you can't forget safety. You have to balance meaningful benefit and safety. And I think, we talk as a community and certainly as advocacy about what is a meaningful benefit. You have to balance all of the totality of the data in front of you. Is this drug safe? If it has a safety pathway, the real world data should really be what decides if it's efficacious over the long term in these long drawn out developmental or degenerative disorders in which in a 1-year or 2-year trial, you may not know. And ultimately it may fail because people can't afford to keep it in a clinical trial. And so you really have to balance, is it safe? Can we get a provisional approval, in which case then we can come back and revisit the information at hand? Does safety still outweigh or weigh against benefit? And I think that's really important for these disorders in which there's no funding to be able to advance them beyond a 1 or a 2-year trial.
And one thing I would add, going back to one of my earlier points around having a treatment effect that is large, I think it's context dependent. And I do see, many regulatory discussions where we get stuck on that piece. How large is that treatment effect? And I think that as we try to contextualize what a large treatment effect looks like, the question of if you're looking at sort of a biochemical change, are you looking at total absolute change, percent reduction? Does it matter where the starting point is for the patient? Those are all factors that I think become really important when we think about that terminology that appears in several FDA guidances around you need to have a large treatment effect. What does that mean? And so really having, sort of the flexibility around that to enable those discussions to advance is important.
Just want to make one more comment too. I do agree. I mean, it's a balance between safety and efficacy, and you're absolutely right. I think the patients need to weigh in from a safety perspective. Like, what are they with this magnitude of effect, whatever it is, because it has to be contextualized. What does that mean for the community? What are they willing? What risk are they willing to take? But I think that needs to be communicated to regulators as well, because I think that it's hard when you look at a review document to understand, but it is meaningful, and they're willing to take certain risks because they see this magnitude of effect to them is essential, whether it is following their parents across the room, but essentially that needs to be contextualized for the regulators in addition, and the safety is a key element.
Great, really important points from all of you. Oh, Becky, did you want to weigh in?
I just wanted to make one brief comment. So great contributions, and I want to emphasize that we certainly appreciate all of the patient voice that is brought to us both at the IND stage and at the NDA BLA stage. I think it's really helpful for, I think, as someone said, to contextualize the findings that we see. Just specifically as a statistician, I wanted to add, when thinking about how to best serve patients, I think there are approaches that we could maybe use a little more to optimize efficiency and making sure that we're using every patient the best way possible. Just one approach is a group sequential type design where we incorporate futility and efficacy stopping rules. And that means that we will stop if the trial doesn't look promising, and continue if there does appear to be some persuasive but not yet convincing evidence to move forward. I think that's just one example where we can use kind of adaptive approaches to really best ensure that we are serving the patients in this kind of internal control approach. So just to add that.
Yeah, I'll just add a few more points as well. So I'm a statistician, so yeah, I really appreciate all your insights and experience. So I guess we have to strike a balance between scientific rigor and what is practical and ethical and the logistic constraint of running a rare disease trial. So like what Becky said, there could be more innovative study design, adaptive design. And I think, so there's also the crossover, like I said, the traditional crossover design that might not feasible for gene and cell therapy trial, but we have like more modified crossover where you have, subjects randomized to gene therapy and then placebo, but then the placebo crossover and get the treatment. So this, but in comparison is still between groups. So this is a more modified, I think, modified crossover design, but patients have all the subjects have the opportunity to receive treatment. And I also wanted to touch upon like what we said about placebo response. It is real in some diseases in the endpoints that you're measuring. So we worked in the file, so where there are multiple approved products, and but there is a high placebo response rate. So how do we design a trial to assess new products, right? You can't, if a product has been approved, it's shown to be efficacious. So if you do it's really hard to demonstrate superiority, and we don't expect superiority over existing therapies. But then if you try to design a non-inferiority trial, it usually requires a large sample size, and so it's with a small rare disease, there's a smaller population, so it's really hard to do a non-inferiority trial, and you have to also come up with a reasonable non-inferiority margin. So in that case, you still, even though there are multiple approved products, you still see randomized placebo-controlled trials where, during the trial, it's a short period of time, so it's 6 months. And so the placebo subjects, all the subjects, they are being monitored during the trial. And if they relapse, they'll be given rescue medication. So for this condition, there is very minimum or no irreversible damage. So that's why it's possible that, that you still see randomized placebo-controlled trial in this particular disease.
Great. Thank you so much. Sadly, we are at time here. There were some questions in the app, and we wanted to get some questions from the audience, but the conversation was obviously so robust and covered so many topics. So I just want to close here and free everyone up for their lunch. And again, thank you to all of our fantastic panelists, and thank you to FDA and Duke-Margolis for convening this really important meeting.
I got it.
Thank you.
All right. Welcome back, everyone. I hope you each enjoyed the lunch break. We're glad you all are back for our next session, where we will now discuss external control options. And we're joined by a very esteemed panel of folks. Very happy that they're here with us. And that being said, I'll go ahead and start introductions. So I am Rachel Hendricks Stirrup. I am the Research Director of Real World Evidence at the Duke-Margolis Institute for Health Policy. I'm based here in Washington, D.C., here with my colleagues. My team spans both Washington, D.C. and Durham at our main campus. So we're delighted to be here to talk about this very important topic that is very prevalent and salient to real-world evidence policy, that is external controls. So I am going to go ahead and introduce you all to our speakers. We have 2 presentations happening before our panel, which will kick off in about 20-30 minutes after our presentations. First, we will hear from Ramona Belfiore-Oshan, who's the director of the Duchenne Regulatory Science Consortium at the CPath Institute, or Critical Path Institute. After Ramona's presentation, we'll hear from Dr. Nicole Mayer-Hamblett, who is a professor of pediatrics and adjunct professor of biostatistics at the University of Washington. Nicole's also the co-executive director of the Cystic Fibrosis Therapeutics Development Network Coordinating Center, which is out of the Seattle Children's Research Institute. So without further ado, I will ask Dr. Ramona Belfiore-Oshan to step up and give us our first presentation for this session.
Thank you, Rachel, and thank you, everyone. Good afternoon. Thank you to the Duke-Margolis team for having me here today. My name is Ramona Belfiore-Oshan. As Rachel anticipated, I'm the Executive Director of the Duchenne Regulatory Science Consortium at the Critical Path Institute. This is a public-private partnership that aims to act as a neutral convener to accelerate drug development. And today I'm here to tell you a story around the use of 2 natural history studies as confirmatory evidence that successfully pushed forward the approval for a drug in Duchenne muscular dystrophy called givinostat, or Duvizat, how the commercial name is. Going back to the rationale of why do we need external evidence in rare diseases. Duchenne muscular dystrophy is a rare disease. And as such, the design of trials when we develop drugs assume the size of the trial needs to be small, the use of placebo needs to be minimized, and yet DMD, as other rare diseases that we discussed today, is a very heterogeneous disease. So how do we know whether a treatment is actually changing the course of the disease? And this is where the natural history come in. Natural history studies that are well developed and prospectively collecting data in a systematic way can provide the longitudinal context and contextualize the history of the disease, providing evidence of what we should expect to happen in the disease course, against which the sponsors can then compare the effect of their drugs. So going into the givinostat, this is a HDAC or histone deacetylase inhibitor that was developed for DMD boys of the age of 6 and above. The pivotal trial, EPIDIS, was a randomized phase 3 trial that was 72 weeks long. And the submission package was delivered to both the FDA and later to the EMA. The FDA provided a full approval in March 2024, and later in June 2025, the EMA, with approval of the European Commission, provided a conditional marketing authorization. As part of the submission package, as was publicly available and released, there was the inclusion of 2 natural history studies as external evidence to support the comparison of treatment effect with the placebo arm of the natural history cohorts. And these were the Synergy DMD natural history study and the Imaging NMD, formerly called Imaging DMD natural history study. To tell you more a little bit about the Synergy Duchenne natural history study, this was a large prospective study that started in 2005. And the tranche of data that was utilized in this case were collected up until 2016. This included a large number of patients, more than 400 patients and families participating to the studies from around the world. And the number of elements, data domains that were collected were very broad. So the power of this data set, and specifically the characterization of many domains of the disease, including cardiac, respiratory, and functional outcomes, were really high. And specifically for the givinostat trial, the use of Synergy was relevant to compare how long it takes for boys with Duchenne to lose key functions such as the ability to perform the four-stair climb test, the ability to rise from supine, and the ability to walk in the 10-minute walk test. So providing this longitudinal context that the trial duration would not be able to provide by itself. Alongside with the Synergy study, the Imaging NMD, formerly Imaging DMD study, was included in the package as external evidence. And this also is a prospective study that started in 2010. This is still ongoing and really capture changes both in the functional assessments of this population that currently also includes other dystrophies, such as the Becker muscular dystrophy population. But for the purpose of the trial of givinostat, it was really relevant, the fact that within this natural history study, it was pioneered the collection of imaging-derived biomarkers, specifically collected through magnetic resonance spectroscopy. And the measure that was captured was the fat infiltration in the muscle of Duchenne boys as determined by fat fraction. So the fat fraction measure in the vastus lateralis, a muscle of the leg, was utilized as an indicator of the disease course and really quantified the changes of the progression of the disease in time. This science specifically informed and guided the trial design of EPIDIS, this pivotal trial. EPIDIS included as the primary endpoint a four-stair climb. The secondary endpoint included both the velocity, other velocity endpoints, and the imaging-derived biomarkers. But importantly, the information provided by the natural history data collected longitudinally across 10 years were able to provide a specific biological window to enrich the population in a way that the trial, the populations were comparable. So this is really relevant because it really signs a landmark for utilization, not only of the functional changes in biomarker, but the biological changes that happen throughout the disease. And using a marker such as fat fraction in this case was very impactful because it's underlying in the cause of the disease that as the disease progresses, you will see very clearly infiltration of fat in the muscle together with fibrosis. So this utilization of biomarker really allow the sponsors to enrich their population, reduce the heterogeneity in the trial, and have also a mechanistic approach to how the drug was changing the course of the disease. So how was this data utilized in the regulatory assessment here? We know that both regulators have stated looking at the totality of evidence, and the two natural history data sets were utilized. And specifically, the FDA stated the acceptance of utilizing this evidence as confirmatory evidence of the efficacy of givinostat. The EMA, as I mentioned, provided the conditional marketing approval, asking for additional data to be collected. But in this case, that could be also compensated by adding patient registries data as part of the future data being collected. So both agencies really see the relevance of adding information such as the external evidence as impactful in the overall decision making. It was evaluated the use of propensity score matching in attempting the balancing the givinostat cohorts with the natural history, and both agencies highlighted potential caveats that come from these methodologies that could introduce bias based on post hoc adjustment and assumptions that are made when utilizing this, the specific methodologies. So what was the quantitative funding of the trial? The evidence show a slowing the Forster climb, so the primary endpoint was met. In the perspective of imaging, we see also slowing fraction infiltration in the vastus lateralis. And overall, when compared to the natural history cohorts, the trial seems to slow the progression 1.6 to 3.5 years. So overall, this package was compelling, and it really shows the unicity of utilizing sources of data from multiple studies and multiple sources indeed. So the use of external evidence here, we can highlight the strength that really comes from the nature of these data sets. These were very long, very well standardized data sets that were collected with a specific protocol, and the longitudinal characterization of both the functional changes, but also the biomarker changes was key to support the proof of efficacy of this drug. No data set is perfect, so of course there were limitations that were highlighted. Among those, of course, the residual confounding factors, the limitations potentially due to the fact that the natural history changes and the standard of care changes due to this potential new therapies available to patients. And so it could become hard to compare the cohorts with external evidence, but the notion is that there might be also challenges related to the utilization of methodologies that could introduce uncertainty when evaluating this data. Overall, the summary message, though, that we take from this specific case study is that really the successful utilization of rigorously collected and timely shared data. I want to emphasize this point because we know data sharing, there was not going to be a potential use of this external evidence. But the timely sharing of this data allowed sponsors to not only use this evidence in their trial, but also use them before as part of the trial design. And this is a very important piece that really gives us important lessons learned for the future. So for future trials, what we can take from this study is really the need for us to pre-specify the utilization of the natural history study we intend to use. It is important that this information between trial and the studies that we select are comparable, and the relevance of use of biomarkers to not only define the functional changes, but the biological changes in the disease, and also as a way, as a drug development tool to empower and the analysis of our trial and the success of our trial through the enrichment processes. So more broadly, what we learned from this is that really sharing data that are collected in large pre-competitive efforts is a very powerful tool that we hope can be sustainable in the future. And so my question to you that I would like to ask you with and leave you with is how do we make sure that this data can be continue to be collected and updated as we move forward with new therapeutics available to the community? With this, I want to thank you all for your attention. It is important to thank the patients and parents and the community hall that succeeded into really allowing this external evidence to be utilized. The sponsors from Synergy, Imaging DMD, and EPIDIS Trials for taking this on board, and all members of the consortia and programs that collaborate in the pre-competitive efforts that really help us refining the path for accelerating drug development through the development of these drug development tools, such as imaging-derived biomarkers. Of course, I also want to thank the FDA for their continued support to the CPAT programs and leadership since 2005. And with this, I'll close up and I'll let the next speaker, and I look forward to a productive panel discussion. Thank you all.
Thank you so much for the introduction today. As was mentioned, that I am co-executive director of the Cystic Fibrosis Therapeutics Development Network Coordinating Center. That is a long term, but we run a coordinating center for CF trials that occur throughout the country and have a large trial network that is positioned to move our pipeline forward. Just waiting on some slides here. Oh, there we go. And thank you again for letting us present our case study today. So very briefly, oops, technical challenge. Yes. Very briefly, cystic fibrosis is a life-shortening, multi-organ disease caused by variants in the CFTR gene. Lung function loss secondary to infection and inflammation is a primary cause of morbidity and mortality. There are about 40,000 people living with CF in the United States, and we've had recent success with the regulatory approval of therapies targeting the underlying cause of the disease by improving defective CFTR protein function. These therapies, termed CFTR modulators, have dramatically improved clinical outcomes for many, but importantly, not all people with CF. There are about 90% of people with CF that carry at least one variant that produces a dysfunctional protein for which these modulators can improve and or restore function, leading to clinical benefit. But about 10% of people with CF are not candidates for modulators due to either genetic ineligibility or intolerance. Some individuals don't even have a variant that doesn't even produce protein for which these modulators could act. So these individuals will require what's called nucleic acid-based therapies as an umbrella term to produce CFTR function back at the mRNA or DNA level. We have a robust pipeline of NABTs, for short, in early phase clinical development. These span both CBER and CDER and include both mRNA therapies, ASOs, and viral vector-based gene therapies. So importantly, these are inhaled therapies. They face many complexities being delivered to the CF lung. In addition, they will require varying redosing frequencies, even for viral-based gene therapies due to lung cell turnover. So because of these complexities, we need multiple therapeutic shots on goal to meet the unmet needs of our population who are not candidates for modulators. We are ambitiously moving this pipeline forward in a relatively small subpopulation of CF. In fact, there are less than 1,000 individuals ineligible for modulators who would qualify for these clinical trials based on common eligibility criteria. In addition, I want to talk a minute about this diminishing participant pool. This is being driven by two key factors for us. The first is that individuals may have limited NABT trial opportunities or exposure opportunities over their lifetime. While the therapies in our pipeline right now are not necessarily considered one-shot therapies, there are concerns for cross-product or re-exposure, especially when moving within and across viral vectors. Secondly, another important note is that individuals in genetic therapy trials in particular are required to participate in long-term safety follow-up studies. This is up to 15 years for integrating-based viral vectors. So while not prohibited, this will complicate the feasibility of someone concurrently fulfilling their long-term safety follow-up for one study while participating in an alternative investigational trial for a second NABT study. Especially if we haven't found that breakthrough therapy right away. They may want another therapeutic shot on goal. Our team has thought a lot about establishing a framework for sequential NABT trial participation in a recent publication that I'll refer you to. However, even despite establishing this framework, we know that we will need innovative approaches to streamline our trial designs in order to enable multiple therapies to move forward in our pipeline. NABT development is fortunately a prime example for us to consider the use of external controls. To begin with, we hypothesize that an effective NABT for CF will produce a robust and meaningful effect on lung function improvement, and above that which is expected based on natural variability. So here in this figure, we've drawn the hypothesized NABT treatment response range benchmarked against what we've observed in prior placebo arms over time. And we've also benchmarked those this range against what we have observed in the past for those modulator therapies. What you will see is that it is slightly attenuated, and this is because of the delivery route. Our modulator therapies are systemically delivered, while these NABTs are inhaled therapies. So we do expect that an inhaled NABT treatment effect will likely be attenuated in comparison to what we observed for those modulators, but nonetheless clinically impactful and above which we observed due to natural variability. So at any point in time, an individual may have one or more NABT trials for which they could consider participation. This is not we are not randomizing participants to these trials. The trial selection is highly individualized, of course, based on willingness to participate. Our network has several outreach and engagement work programs going on that I don't have time to speak about today. However, trial selection is, of course, determined by eligibility. Some of these therapies are variant specific, such as ASOs, others are variant agnostic. Risk-benefit discussions with their providers, which will vary across these different modalities. But quite honestly, practical considerations. Where are the trials being run in relation to where they may reside? Importantly, it's not feasible necessarily for a single trial site to run all of these clinical trials due to the intensity of running these genetic therapy trials and mixing in inhaled delivery. We hope in an ideal setting that all of them are going to move to later phase development. Of course, an ideal situation is that they would go into each of them into randomized placebo-controlled trials, but we know this is not feasible. So essentially, we are working ahead to build an external control repository in partnership with the Critical Path Institute and their data sharing platform to be able to share this repository across our industry sponsors, to enable the design and the development of either hybrid control trials or single-arm trials that would reduce the need for or size of these placebo groups across the pipeline. And you'll see here, this is a two-way street, and that this placebo data can contribute to that repository over time. As we build this repository, we not only have data from our natural history registry that has existed for many years. At our coordinating center, we also have archived past trial data. But we're taking a more proactive approach, and we have designed a fit-for-purpose prospective control study to collect control data on the population who are not candidates for modulators. We actually have a window of opportunity here of time before these trials move into later stage development to collect this data in a more intentional fashion on a relatively NABT naive patient population. We have standardized the outcome measures and timing aligned with what we anticipate our later phase NABT trials will be. We are collecting comparative safety data in addition to efficacy data. Conducting this in a regulatory compliant fashion and conducting it in the same trial network as the genetic therapy trials themselves. Importantly, this is also an opportunity to engage our community in research, some of whom have not participated in trial before, so we have an opportunity to engage them in research, perhaps even before approaching them to participate in an NABT trial. So I know what you're thinking, is this a master protocol? Well, I like to refer to this approach as a deconstructed master protocol, whereby individual sponsors will run separate NABT trials, but utilize a shared control arm. So the advantages of this approach is that it does really solve our most critical problem in terms of decreasing trial sizes and enabling a strategy that promotes more innovative designs that will enable us to move more shots on goal forward through the pipeline. It also, as a patient organization, does not require us to have an exceptionally complex master protocol with multiple INDs. And for this class of therapies, genetic therapies, it is quite complex that would need to be negotiated across competing sponsors and could ultimately slow us down. However, we do have challenges to address. There are multiple, and I'll just highlight, the FDA's guidance on external controls, because that's really the heart of what we're strategizing here and promoting. Of course, there are several concerns that we must address. One could argue that by design, the REACH study is meant to mitigate some of these concerns, but we know we are going to have to address selection bias, unmeasured confounding. And in addition, outside of master protocol, we must identify standardized methods that can be used across sponsors so we can compare apples to apples in terms of treatment effects. So not only are we investing in control data repository, but we're also investing in methods work. Statisticians from our coordinating center, our clinical trial coordinating center, are partnering with a statistical team at Telperion right now to identify efficient and robust methods to incorporate external controls in our future NABT trials. We have designed a number of different simulation studies that we are launching to really evaluate the performance of methods for our specific situation, our disease, our indication, and our outcome measure, covering the trial sizes that we think will be the target trial sizes that will be feasible for later phase, the treatment effect ranges we are anticipating, and then covering a number of different statistical methods, including Bayesian and frequentist methods. These methods here listed are a little bit more conventional in terms of testing for the average treatment effect. Average changes in lung function is our primary efficacy measure. But we're also stepping back and playing around with the hypothesis a little bit and looking at methods that extend the randomization inference framework to hybrid control trials in order to test what's called the sharp null hypothesis of no treatment effect. Because we have to test the performance of these methods in a pretty small sample situation. So I'll just show you, this is the first time we're showing data today. No, it's not the first time, but some simulated data here on methods, actually. So here's some data from initial round of our simulations. And what's shown here is a comparison of the baseline characteristics between what we're using as our external controls and the black boxes here versus about 2,000 simulated trial data sets in the bubbles across various baseline characteristics. The main point here to see here is that they vary, they differ. So for baseline lung function, our external control population has a slightly lower baseline lung function than our trial population going into these simulations. This is probably real life, especially as we're pulling in external control populations from various, registries and so forth. We need to pressure test these methods, knowing that, these populations will likely differ. So here are just some preliminary results from a few of these methods here. And what you'll see, this is very relevant to some of the panel discussion earlier today, is that you'll see that the bias in estimating the change in lung function is actually quite low. We have a very good accuracy with these methods, but it's the power, it's the efficiency that takes a hit at these smaller sample sizes. So I was happy to hear that there may be some flexibility in that, and that there may be more of an emphasis on accuracy, knowing that efficiency is going to be an issue. However, for us, we do have one candidate method, Bayesian and additive regression trees, that is showing quite promise. It may be performing better because it doesn't force a linear relationship between baseline covariates and outcome measures, for instance. But this is really just the tip of the iceberg in terms of what we're doing. We have more methods on the docket that we're evaluating, including some of those Bayesian borrowing methods as well. So stepping back, my goal today is just to present our approach, which is really a sponsor agnostic strategy to meet our urgent unmet need. Not only are we building an external control repository that can be shared across our sponsors, including the collection of fit for purpose data in our control study REACH, but also doing some independent methods research to tee this up for our sponsors and really look at what's going to be optimal for our specific outcome measures. And we just want to end by thanking the organizers for hosting the RISE workshop and the series, because it's really this collaboration with the Rare Disease Innovation Hub that allows us the opportunity as a patient organization to move this sponsor agnostic strategy forward. Some of the other mechanisms do depend on holding that IND, which we don't. And so this has really been a phenomenal opportunity for us to engage across the agency. So thank you.
Wonderful, excellent presentations. Now we are going to move into our panel discussion. So I'm going to ask our speakers to join us on stage with a few additional individuals. So we have joining us for the panel discussion, Dr. Najat Bushkush, who is a pediatric hematologist oncologist who serves as an associate director for pediatrics in the Office of Clinical Evaluation, or OCE, at the FDA's Center for Biologics Evaluation and Research, or CBER. Next, we have Dr. Scott Demarest, who is an associate professor in the Department of Pediatrics in the Division of Neurology. Next, joining us is Tracy Sikora, who is the Vice President of Research and Clinical Programs at the National Organization for Rare Disorders, or NORD, where she leads NORD's Rare Disease Centers of Excellence Network and the I Am Rare Patient Registry Platform, a research granting program. And then we have Dr. Arup Sinha, who is a senior staff fellow in the Division of Biometrics, Office of Biostatistics, which supports the Office of Oncology Drugs in the CBER office at the FDA. So without further ado, I will go ahead and make my way this way. So we are thrilled that you all are here. And the presentations were excellent. I was able to take a lot of notes with some key takeaways of really things that we can consider in this discussion today. But what I want to do is, just first kick off this discussion with something very general, perhaps for each of you to respond to or chime in on. Just give us some of your reactions. So considering what we learned in this, in the prior presentations, work that you all do across your institutes and centers, some successes, some, arguably failures that are more like opportunities. Thinking about all of that, what are some of the most successful examples that you can think of around external control use? And more importantly, how can lessons from those successes become extrapolated in other cases or be applied elsewhere? Just generally, what has been your experience? And whoever would like to go first. I know we heard from our presenters a couple of examples already, so I think I'll turn it to our discussants. And
Sure.
Arup, go right ahead.
I can go first. Hi, this is Arup Sinha. I am a statistician at the Center for Drug Evaluation and Research. I support oncology, a division of oncology 2, which mainly reviews thoracic, head, and neck cancer. I also closely work with the pediatric development programs that gives me the opportunity to work with rare diseases. And I'm happy to share some of my experiences with you and learn from you today. So successful stories, actually, I'm very excited to see that over the last, I joined FDA in 2018. And over the last few years, I have seen several, like more and more external control trial, or real world data proposals coming in. We have some successful stories. Initially, when I started, there was an approval. It's selumetinib for neurofibromatosis 1, who were symptomatic, inoperable, who have symptomatic, inoperable plexiform neurofibromas. And in that trial, the selumetinib was approved based on a single-arm trial using overall response rate endpoint. And who are not working in oncology, response rate usually is measured through the tumor shrinkage. So though this was approved based on data from a single-arm trial, the applicant had submitted information from a natural history study, which was conducted by NCI. It's called NCI Natural History, I think NF1 study or something. I forgot the exact name. So what happened, we had looked at the data of the natural history of the plexiform neurofibromas, right? So we did not conduct a formal comparative study, but what helped us is it gave us valuable context to characterize the data, the responses we observed in the single-arm trial. So that one is, and the other one I would really like to mention, it's a recent one. And this application had used external control trial for comparative efficacy. And this indication was DFMO treatment for high-risk neuroblastoma. And this trial used a single-arm trial data where patients with newly diagnosed high-risk neuroblastoma, they received their induction therapy, consolidation therapy, maintenance therapy with immunotherapy, and then received DFMO. And patients, usually this induction consolidation and maintenance therapy, that's the typical standard therapy these patients receive. So this single-arm trial compared data from a previously conducted randomized trial where patients received these 3 phases of treatment and went on surveillance. So what they compared patients who received 3 phases of treatment and received DFMO versus patients who received 3 phases of treatment and did not receive DFMO. So one of the key feature I really highlight is the sponsor communicated with us from the very early in the design and analysis of this externally controlled trial. And FDA's consideration or evaluation, this data was considered fit for purpose. Having fit for purpose data is very difficult. So it was a fit for purpose. So we were able to apply a lot of statistical methods. And finally, the drug was approved for this indication. And the reason I brought those 2 examples, one is when we think about externally controlled trial, it does not necessarily have to be always a comparative study. We can use data supportive from the external control data to characterize the data we have in hand, and that can help us to evaluate the results of the current data we have in our hand. So that's it.
Awesome. Go right ahead, Scott.
Very interesting. So I'm Scott Demrus. I'm a pediatric neurologist and an epilepsy doctor by training, and have been involved in a variety of natural history efforts with the goal of hopefully one day being used as historic controls in disease-modifying trials. The example I'm going to point to of a success was something I wasn't directly involved in, but have seen the impact of within my clinical practice. And maybe a bit controversial as a success story. It did lead to an approval, but as I understand it, it was actually not a very easy approval. It was a difficult one to get across the line. And that is cerliponase alpha for CLN2 Batten disease. This is an enzyme replacement that is given every 2 weeks via an intraventricular access device to prevent degeneration and death of a terrible early onset neurodegenerative condition. I think what's interesting about it, it did use historic controls in the process of getting FDA approval. There was actually quite a stark difference between those historic controls and what was seen in the trial on a relatively blunt instrument that was being utilized. 2 different subscales, which each had 4 points to them, right? Like we're not really talking about a particularly robust outcome measure, but it was one that had been used and validated and that there was historic control data associated with. And in the process of now implementing that treatment at our institution and working with others around the country and internationally that utilize that treatment, we are able to say that there's probably the rate of degeneration was cut to probably something like 20% of what it was prior to that treatment. In other words, if the degeneration that occurred over 2 years probably occurs over 10 years. I mean, I think like we can all agree that this is actually a home run, right? This is an incredibly effective treatment that we've been able to see in terms of following these patients since it was approved in 2017. But actually getting that there and why it was difficult to demonstrate that within the context of a clinical trial, I think it's both a success and maybe an opportunity for learning and understanding and how we could avoid it being challenging for something that is so obviously beneficial to be difficult to get an approval of in future trials.
Yeah, it's really hard to top that. These are wonderful examples we've heard from so far. I'm Tracy Sikora. I'm the Vice President of Research and Clinical Programs at NORD. And when I think about success in this area, I do think about these great examples that we've heard from about where we get to approvals. But sometimes I think, what does success look like a step before that? How do we even successfully get to the area where we're collecting the data that we need? And it makes me think back on my career. I've been in rare diseases over 15 years. I've had the privilege of working on many different rare diseases. And one of the things that I think really is a predictor of success for the diseases that move forward through the drug development process is something that maybe I would term patient-centered collaboration. So I talk about collaboration because when I think about external control arms, I really think of that as one of the areas in drug development that's really collaborative. You know, we've seen how groups come together, they share data, they work in a pre-competitive space, and I just think that that's incredibly important. But I also think about that patient-centered part of it. So, you know, at NORD, we have a patient registry program, and we consider that these patients and these patient advocacy organizations are not just users of the platform, they're actually the owners of those data, they're owners of that registry. We heard from some great speakers earlier today who spoke about how patients, they're smart, they're knowledgeable, we can trust them to actually work with others in this drug development space. So we can trust them to own these registries themselves. So our platform really empowers patients and patient advocacy organizations to do that work themselves. So I think about, you know, these groups don't have to be at the level of sophistication where they're running their own amazing collaborative work like we've heard from today. They can come to us not necessarily knowing exactly how to work with an IRB or exactly how to write a protocol. And they can work with organizations like NORD to learn that process so that they can actually become the owners of those registries. And I think that that patient centricity is incredibly important to getting the data that's actually usable, so that we can see these successes that we've heard about. So this is not the only model that works, but I think that including patients early and often doesn't just mean they are have a seat at the table. They're not just on an advisory board. They're actually developing the natural history studies and the registries where we're actually be able to use those data later on. So it can't just be early and often. It has to be that we empower and trust the patient organizations and the patients to be true collaborators in this process. I think that's really where we can learn some lessons and see some successes.
Wonderful. I think we
Maybe I can go.
go right ahead. Yes.
Yeah. So I'm Najat Bushkuj. I am the Associate Director for Pediatrics within the Office of Clinical Evaluation, within the Office of Therapeutic Products within CBER. By training, I'm a pediatric hematologist oncologist. I've been at the FDA. As of today, it's been 9 years. So I've gone through, several years of changes and really good changes. And I witnessed them on hand from being a clinical reviewer to then becoming overseeing the product developments in pediatrics and rare diseases. And I would say, I can give you several examples of success stories. They're mainly centered towards communication and meetings with the FDA and patient advocacy groups early and often. And I can't, emphasize that enough. I can probably just, mention the Kymriah story, which is the first chimeric antigen receptor therapy for pediatric leukemia, which was approved in pediatrics in 2017. And I really admire that story because it was the first pediatric approval, even in a disease where adults are existent with the disease. However, the therapy was approved in pediatrics first. This was based on single-arm trials, same as was mentioned before. The response rate was really the basis for approval. The other data from historical controls was used in that setting to support the approval in terms of efficacy. There are several gene therapy-based therapies that were approved. Zolgensma is one of them, which is an intrathecal gene therapy that is administered in patients with SMA, which is a type of severe neurological devastating disease. There's other gene therapies in the pre-symptomatic or early stage of disease that was approved. Lenmeldy, for example, where it's a gene therapy that's administered to patients with really early in the disease setting to prevent the devastation of the disease. And both of these, Zolgensma and Lenmeldy, they were approved based on single-arm trials with supportive evidence from national history studies that served as historical control. So again, I think, I can give several, successes, but, in all of these cases, the sponsors came to the FDA early, had several meetings, trying to design their studies and get kind of an agreement with the FDA and with the review team on how is success going to look like and what data are needed to support approval. Again, I think as was mentioned before, the external controls can be used not only to support efficacy, however, to support safety as well. And they can, either be used in the pre-INDs, during early stages of drug development as well as post-approval to capture more data and serve as repository of valuable information that can be provided by patients to support the long-term safety and evaluation of these therapies. Maybe I'll stop here and
If I might add, I think one very important point is really not targeting just the therapeutic approval process, but going early in the pipeline. To me, success is really utilizing this data also to optimize the trial design, optimize the selection of the endpoints, the size of the trial, the length of the trial. And so this data are really able to inform the trial design before we target the actual regulatory pathway. And so to me, the success is really when the communities come together to build the infrastructure so that this data are shared And we removed the silos that will limit us to actually access this data that inform both the trial design and the drug approval.
Absolutely. I don't know if you have anything else to add, but I'm definitely hearing a thread that patient-centeredness is key, especially as we think about, Scott, as you mentioned, percent improvement in patients' ability to function in the real world, and demonstrating that improvement using real world data that is in the form of external control data. It's hard work. It's not easy, but it's worth it, especially when you do it in a way that is very patient-centric and focused and meaningful. But I don't know if you had anything else to add in reaction to or in addition to what our colleagues have said today, but I'll stop to give you a moment if you'd like.
I think what I was just going to mention and highlight is that, of course, there's been numerous examples, and I think we're seeing the success of that come through with even the announcement this morning from CEDAR of a more formal acknowledgement and recognition of a new a pathway that recognizes one confirmatory study and then the supportive evidence from external controls. And that is really a step forward, is a priori having that pathway available, especially from the rare disease setting where we are working with developers, investors, and it's our responsibility to show, there is a pathway forward, even for rare disease, utilizing these other mechanisms. So that formal recognition of these pathways is really so important.
Yes, absolutely. And, Nicole, you mentioned during your presentation that, a sponsor agnostic strategy, might be useful, especially in the context of a master protocol, so to speak, that would involve the integration or implementation of real world data in study design. And so you also mentioned a shared control data repository. I want to take a moment to, talk a little bit more about that, especially in the context of, what could be successful, what based on lessons learned that you all have, discussed today or presented today. What should we think in that regard, where, patient-centeredness, again, putting that front and center, where patients will come together to ask, can we create some sort of repository like this to inform several sponsors that are working to develop therapies using our data, our externally controlled, hopefully longitudinal natural history data, to meet us where we are. What are your general reactions to that idea or to this mechanism of going about, again, patient-centeredness, meeting them where they are, meeting them to the occasion where they are coming to you and saying, here is our data, here's our information. Please tell us what we can do about that. Tell us how to build the infrastructure, improve that infrastructure to make this meaningful and worthwhile. What are your reactions to that approach based on where you sit within your organizations and within the science of what we're doing here?
Well, maybe I can start. I think the amazing work that's already been done, we would love to replicate that. So at NORD, we represent every rare disease. It's a lot of rare diseases to represent, but the more we can do to lower that barrier to get from, you're an ultra rare one person, you're one mom, then you're trying to find the others, to something like what you all have built out. What can we do to make it so it's not the longest journey every time, that we can shorten that timeline to go from, starting to thinking about research to being able to build that registry or build that coalition. So I think it's a lot about infrastructure building, but earlier today, there was some talk about archetypes and doing crosses, and I think this is where it's really about. It's really about taking lessons learned from one disease that may not even be related to another disease and sharing it. It's really more about the systems and the infrastructure. So any lessons learned, I'm going to be following up with you later to hear more, that we can share with other groups. I think that's really where we see success.
Yeah, I couldn't agree more. So I think that there's opportunities to solve some of the challenges around rare diseases when we approach them in this way. And I totally agree with this concept of rationally grouping. I'm in the process currently of trying to stand up what's a consortium agreement across a large number of rare diseases to support natural history for developmental and epileptic encephalopathies, or DEEs, which has about almost 1,000 genetic causes identified so far. So, we can't do all 16,000 rare diseases at once, but if you can get 1,000 of them in one go, like, this is starting to actually be scalable and maybe feasible, right? And what unifies them is the presentation, the phenotype, the medical challenges. Not because every patient has every medical challenge, but because the range of medical challenges across that group is similar enough that you can design a platform protocol, a basket protocol, we call it whatever we want, a master protocol, but you can collect those things in a feasible way with high quality, robust data for natural history purpose. And lots of different things could be utilizing that historic control group in various ways. So it may be as a comparator, it may be as a comparator safety, it may inform design of a trial at a minimum, but you can do that in a basket way. So you have scalability, you have feasibility, you have the data aggregated and not siloed away from various groups to be able to use it, or different companies to be able to use the same data set. And that's really addressing a number of the fundamental challenges that you see in rare disease in one overall protocol that is pursuing that. I mean, I think Children's Oncology Group is probably the first group that really successfully implemented this type of structure where you could have iterative learning in this way. I'm just trying to learn from people who have done it well and apply it in a different aspect of medicine. But I think that's a critical component of this is you batch the infrastructure and then you iteratively apply that across different treatments so that you move the bar collectively very far, even if each treatment only moves things a little bit.
Go right ahead. I was just going to step back and say that I think we have a tremendous responsibility to ensure that every single, I'm going to say data point, is maximally used in this area. And I love the conversation and the point made this morning that it's not a data point, right? It's somebody who has come in and invested their time, their family has invested their time in providing us this precious data. And so we have that responsibility to really maximize the use of that in whatever way possible. But to do that requires a tremendous amount of intentionality too, in terms of how we can repurpose that data for all of these different applications. We are looking at making sure our informed consents are as broad as possible, but yet informing our individuals that yes, these data could be used for commercialization purposes, which is not typical, especially for academic studies. I think that's not been the standard, but we really need to think far in advance. We're working with our industry sponsors to provide the potential to, and through their consent forms to be able to link with our patient registries over time. Maybe that could provide some internal control data that they may need for their development programs. And so we've developed consent language and provided that to our sponsors and say, hey, you might want to use that. So that's sort of, the exercises that we're going through in order to really maximize, build, not just the control data itself, but the potential to use what we were already collecting.
Yes, go right ahead. I can also just add in, I can't agree more about what you just said, because, just collecting data, it's not sufficient enough. You want to make sure that you're going to use that data. It's valuable. If you discuss with regulators early enough, you know what to capture, how accurate the information that you need to capture needs to be. You also would want to make sure that you capture the correct outcome measures that are going to hopefully be useful later on for a marketing application or to help inform drug development in terms of supporting efficacy and safety. So it's very critical to, as academicians and sponsors and patient advocacy groups to work together along with regulators to come up with exact data points that are going to be useful later on. And that's going to depend on the disease characteristics, patient characteristics. There's a lot of heterogeneity in general, especially in the rare disease space, ultra rare disease space. So the information that you're going to capture needs to be, just discussed with the regulators early and often enough so that it's very useful.
Yes, early and often engagement is something we hear a lot, and I think that will continue moving forward. But thank you, Najat, Nicole, Tracy, Scott. We do have a question from the audience for Ramona. And I do want to remind everyone who's with us online or here in person, please feel free to bring your questions to the panel if you have any. We'll try to get to them as we have time to do so. But Ramona, we have a question from the audience. They mentioned that you spoke on limitations of the natural history study approach, including evolving treatment paradigms. What time frame should future studies consider in that regard?
Well, in general, I think this is something that we're still learning because as the community access to new drugs, we are learning what are the potential effects into the natural history study. The key component here to define our timeline is to ensure that the data currently collected are shared back with the community of researchers as soon as possible. So at this point, really, it is complex and potentially not feasible to make an assessment of the timeline. But what we can do is certainly act ahead and make sure that we are looking at this data very carefully with different perspective and expertise to ensure that the way we are collecting the data and new natural history studies can be also utilized for trials in the future where we have potentially multiple natural history, multiple standard of cares that will be characterizing the population. So playing ahead, now that we have this knowledge, it is very important. And we certainly don't want to start from scratch. We want to learn from what we've done so far. So adding information from the real world and ensuring also the rigor of collecting data from the real world is matching what's needed to reutilize this data for regulatory purposes, because we know that the nature of different sources of data might change. And so having standards for data collection that allow us to both collect data, but also translate those to uses that can advance further drug development would be really, really important as we move forward.
Yes, definitely. You mentioned something very interesting where, especially in the rare disease context, that's a global phenomena where, rare diseases can show up across the entire globe, across every country, every continent, and making sure that you account for heterogeneity in the standard of care, across all of those regions, taking that data coming from each of those places to inform a single repository, to drive a singular purpose that is, treatment development. That is something that you have to reconcile, again, in partnership with patients and as a sort of custodian of their information, their data to do that. I think that's a really important operational consideration, and really a real world phenomena that shouldn't be underestimated. And I'm glad you talked about that to some degree just now. Going back to our audience questions, I do want to encourage everyone to join us, those of us online and here with us, join us at slido.com. At the QR code that you see here on the screen. Feel free to type in any questions you have for our panelists and presenters. I'm going to move to another question that we have from the audience. Someone asks, any experience among the panel on a centralized global strategy for rare disease where data and design has been accepted across the FDA and the EMA, for example, or that is the European Medicines Agency? Are there discussions between the FDA and the EMA and other regulators on rare disease clinical designs that are suitable for regulatory approval? So Art, this might be a good question for you. I'll pass it to you.
Okay. Well, first, I think I didn't mention in my first comment that my views are my views, not FDA's. And I don't want to go further to EMA. So there are collaboration between FDA, EMA, for example. This is, you can find it in the FDA website, like Project Orbis, where drugs are evaluated kind of contemporaneously in multiple countries. And so if we are evaluating a drug around similar time, there are some shared experiences on the review process. Other than that, like in particular in rare disease, I probably would not be able to comment in that particular, but there is a mechanism where drugs are reviewed globally, around the same time at the FDA.
Maybe I can add in as well from the clinical standpoint. We do have as clinical reviewers and product reviewers and preclinical routine conferences and meetings with the international regulators. We have standing meetings once monthly for the oncology products and also for the advanced therapeutic trial ATP with the EMA and other international regulatory agencies. We do have regular meetings that happen almost monthly. Also in the pediatric space, there is the pediatric clusters meetings with international regulators, where if there is a sponsor who's developing a product for a pediatric indication within either CBER or CDER, that can be discussed with the EMA or other international regulators in terms of overall drug development, endpoints, outcome measures that are going to lead to the approval of certain applications. So those are also on a monthly basis. And they're conducted under confidentiality agreements between agencies.
Okay. Very much. All right. Well, again, we encourage questions from our audience members as we move forward with the panel. We've got about 10 minutes left. So for those of you who might have burning questions, please feel free to ask them sooner than later as we prepare to close out this session in about 10 minutes. But I'm going to go ahead and just pose another question for the entire panel. So what comes to mind as far as potentially newer innovations that might hold promise within the use, or regarding rather the use of external controls? So we talked a little bit about innovative methodology that, or even existing or underutilized methodologies even that coexist with external control trial approaches. What comes to mind when you think about innovation and externally controlled trials or external controlled data? Just off the top of your heads, I'll pass it to each of you. We'll probably go in order here, but yeah, go right ahead.
From my experience, I have been seeing that the sponsors are proposing some innovative trials, including hybrid control, right? So even within a randomized setting, unequal allocation, to enroll more patients in the experimental treatment arm, then you can augment the control arm with external control and some, Bayesian methodologies, including borrowing. So those are some proposals we have seen, and I think those probably are and user propensity score. I think one of the important thing to mention that when we are using external control, it is super critical to find a fit for purpose data. If patients are not comparable, no statistical method can salvage that. And so user propensity score methods and or some other methodologies helps us to balance the patient population. So we have seen user propensity score, either, you can name it ATT or ATE. So those are some Bayesian methodologies, propensity scores, augmented or hybrid controls.
Yeah, I think, I don't know if it's particularly innovative, but I think it is a way of applying existing principles. One of the challenges is to make sure that the data is super high quality, whatever you're using for historic controls. And there's been a lot of efforts around ways of trying to do that are more cost effective. I think at the end of the day, you want really high quality data, it's going to cost a certain amount of money. So I wonder about whether you scale by having baskets of rare diseases that are grouped in rational ways, rather than by focusing on reducing the cost of data collection, so that you can ensure that you have very high quality data. And I think a lot of times we struggle with how do we group, what makes sense to group together. And we often will group by biological mechanism of disease, which may or may not make sense. But ultimately, in terms of the quality of data that you can get, It's based on the symptoms of the patients involved. And so you may have very different biologic processes, different genes and things that are leading to convergent medical symptoms. And if you can capture those accurately as a basket, it doesn't matter that they have different biology behind them. The therapeutic approach may be different ultimately, but for historic control purposes, you can still aggregate those patient populations of rare disease in a way that allows you to scale and bring cost effectiveness to the historic controlled data set that you're trying to develop.
Interesting.
Yeah, I mean, I think I have a similar answer. I think I'm going to say I'm really proud of this group. We've gone all day without mentioning AI, and I don't want to talk about it either. I'm proud of us. But there's I'm going to use some euphemisms. I'm going to say technology, there's innovations, there's advancements we've made that make the collection of data hopefully more higher quality and faster and better and more scalable, right? So I've been following this for many years before this AI boom, and it's always that technology is always a few years away from being able to go, from the clinician collecting really high quality data at a clinic visit, just as part of their standard of care, potentially using some type of technology, all the way through when we're looking at, external control arms include things like real world data, so that when we know in rare diseases, we go to these big data sets, it's a mess, it's hard to use. Is there a way that we can find better ways to clean those data in a scalable way? So I think, it's not there's a lot of people working on this, and now more than ever, this technology is advancing really quickly. It does still feel like we're a few years away, as we always have been for many years now. But I think that it does hold a huge amount of potential, both very early on, right? And that the patient is being diagnosed and we're collecting data just as part of the clinic notes being dictated or being coded in a certain way, all the way through to how those data are actually utilized and patients are found. And so I think that we're not there yet, but there's so much innovation being done here that we're close to seeing some real breakthroughs there.
Indeed. Awesome.
The first session today talked about the deep statistical toolbox that is developing, and I will say, and the need to kind of get out of our comfort zone. And I'm feeling that, as a statistician trying to learn these methods that it's so vast, and that we can go beyond propensity score matching and tap into them. And it's going to be one method does not fit all approach. I think the optimal method is going to differ by disease, indication, and outcome. And so we're going to have to invest a lot, I think, to really evaluate that, to be able to optimize that for each application. I was also going to touch on AI a little bit. This is beyond my level of expertise, but I do think that there is a potential role, even bringing it into the methods for estimating treatment effects. Think about the complexity of the association between our baseline predictors and outcome. It can't all be explained in a linear model, and that's what a lot of these methods do. Or the performance of these methods may vary depending on how well we can describe that association. We very well may be able to describe it better using methods such as AI to incorporate as part of this. So I think some open-mindedness as we move forward and getting out of our comfort zone could really help us here.
Well, I do have a case study that comes from the Duchenne Regulatory Science Consortium regarding the generation of novel methodologies for utilization of data that comes from different sources. We developed disease progression models that describe the disease progression in Duchenne based on 5 disease endpoints that characterize the functional abilities of Duchenne boys, as well as their respiratory abilities. And these disease progression models, which utilize model-informed drug development methodologies, so clinical pharmacology, utilized to define the quantification of how the disease changes in time. These were utilized to then build a clinical trial simulation tool that then we published for the entire community to freely utilize this tool as a first approach to the trial design. And so going back to the discussion of the power of using external evidence earlier in the pipeline, in the development pipeline, these tools can be utilized to define trial endpoint selection and by modulating different characteristics of this Shiny app or the R package where sponsors can upload their own data, it is possible to simulate in silico different scenarios of the trial performance and have an idea of what would be an optimal design. So this is an example of utilization of novel methodologies. And interestingly enough, this was also utilized by the sponsors from Givinostat to then look at some of the long-term predictions regarding FSE and Forster climb as the endpoints on their trial. So this is certainly an example for the community to extrapolate the need for data sharing. These disease progression models are built based on 18 data sets from different cohorts of different natures. So you can imagine how the power of analysis is augmented compared to running the same analysis in a single data set. So this community effort was really key for us to have the success, and we look forward to include more data as the natural history changes, because these models are trained on the data that we utilize to build them. So this is also a way to update these adaptive tools as the time goes by and learn more about the disease and how the disease is changing in time.
Sure. And final word now that we're right on time.
Sure, maybe I can just put a plug-in into the word extrapolation, which can be the data that are generated from external controls can be leveraged to be used to expand indication or get new indications in certain populations that was not studied. For example, if a drug is was only studied in a pediatric population, the data, the external controls, the natural history, the real-world evidence can support expanding that indication based on several factors. And I would encourage you all to refer to the extrapolation guidance, the ICH 11 guidance, where they, if the disease is expected to be similar between, two different patient populations, let's say adults and pediatrics, if the product's mechanism of action is expected to work the same, then data from certain population can support expanding indications to other patient populations.
Excellent. Well, Nishat, Ramona, Nicole, Tracy, Scott, Arup, thank you so much for joining us for this panel today. It was wonderful hearing from each of you and all of you.
Thank you, everyone. Welcome back. My name is Steve Berman. I'm the Vice President of Science and Regulatory Affairs at BIO. And we've had quite the day so far, and it is my great pleasure to be able to moderate this distinguished panel to help us wrap up the day. As a reminder, we have a Slido available for anyone who wants to ask questions to the panel. Please go to slido.com and type in #RISE, and you can submit your question, and we'll address it if we have time. But like I said, we have a great panel. We heard a lot of great discussion today. And so let's get straight into it. I'll begin by introducing the panelists just in the order that we have here. So first we have, starting at the end, Dr. Vijay Kumar, who currently serves as the Acting Director of the Super Office of Therapeutic Products, OTP, in CBER of the FDA. Next, we have Dr. Mark Levinson, who is currently the Deputy Office Director of the Office of Biostatistics in CDER, the Center for Drug Evaluation and Research at the FDA. Next, we have Dr. Samia Al-Zaidi, who is the Global Head of Clinical Development for Gene Therapy Neurology at Alexion Pharmaceuticals, AstraZeneca, followed next by Dr. Elizabeth Berry-Kravis, who is Professor of Pediatrics and Neurological Sciences at Rush University Medical Center in Chicago. We have Annie Kennedy, who serves as the Chief of Policy, Advocacy, and Patient Engagement at the Every Life Foundation for Rare Diseases. And we have Mary Dwight, who is the Senior Vice President and Chief Policy and Advocacy Officer for the Cystic Fibrosis Foundation, and who directs the foundation's public policy agenda and Compass, a team of CF patient advocates who provide assistance services to help navigate insurance, financial, legal, and other complex issues. So we have a great and diverse panel here today. And I am very excited to be able to help moderate the discussion. Like I said, here is our Slido code if you'd like to participate in the discussion. What I'd like to get started with are just observations from the day. Maybe this will be an opportunity for us all to share a little bit about who we are and the perspectives that we bring to rare disease drug development. And so for each of us, please just introduce yourself and then tell us, based on the discussions today, what are some topics that really stood out to you from the discussion? So Mary, why don't you get us started?
All right. I'm Mary Dwight. As Steve said, I'm from the Cystic Fibrosis Foundation. And I just celebrated my 19th anniversary with the CF Foundation. So it's always a time for reflection for me on the point of my anniversary. And it's been a remarkable 19 years in cystic fibrosis. I actually met Amy, I think, very, very early on in my career. And today has been interesting because I think probably the first time I met Amy was at a meeting where we had a lot of the same conversations about the importance of patient voice, the uniqueness of data in rare disease, and the challenges of adapting clinical data into a rare disease setting. So in some ways, today has been familiar, but I also think it has been both in the story of cystic fibrosis and also what we heard in many of the case studies and examples and case stories of success today, a time of optimism. In CF, I think we have been an example in rare disease of how you can really leverage the patient voice, not only their data, which has been critically important, as you heard from Nicole in the last panel, but also their charge to us on what matters to us, what should we be measuring, and then how do we use those calls of tell me this is what is meaningful to me, how do we translate that into data that can be used in a regulatory setting? And we've seen great success in many symptomatic approvals at the FDA over the years, but also very meaningful modulators that address the direct cause of the disease. And it's really transformed the disease for many people with CF, although not all, as you heard from Nicole in the last panel. So I think I've been pleased with the recognition of the uniqueness and the heterogeneity and the complexity of rare disease and the enthusiasm that we heard today for a lot of new approaches. But the one thing I'd love for us to keep talking about is the value of data sharing. That was a consistent theme over the day. We heard it from certainly the patient voices at the table, but also the regulatory voices and sponsors. And we need to keep exploring that because especially in rare disease, especially as we get into ultra rare populations, we are going to be able to have meaningful impact much faster for the people who need these new treatments if we steal shamelessly and share seamlessly. And I think we do still have a way to go on that.
Thanks for calling out the importance of data sharing. Annie?
So hi, it's wonderful to be here with everybody today, and thank you to Duke-Margolis and especially to FDA for bringing us all together. We were a part of the community that advocated for a very long time for the existence of the Rare Disease Innovation Hub. So I woke up this morning a little bit like it was Christmas morning that this workshop was happening. My son was like, what is happening? But this is really an exciting day. So I think just a few themes that for me today resonated, and like Mary, there were things that were common themes that I felt for a very long time in rare, but was reminded of really throughout the day. One is that we have sophisticated, savvy, amazing patient advocacy organizations within the rare disease community, and time again today, we heard examples of that. Just leaders from the community who talked about the capabilities of the community through the natural history studies that are being driven in rare, patient preference studies, efforts to help develop endpoints and outcomes in rare disease. So many examples of that. Equally, the dedication of sponsors in rare disease was talked about and featured throughout this. And then we saw so many different models that are really bringing that to bear in rare disease. So that was really gratifying. I think one of the things that is really important as we think about the opportunity of the Rare Disease Innovation Hub and sort of the context for what has been happening in rare for so long, is that sort of as I was driving here today, I was reminded of when I was little, I would go to summer camp. And one of the things that would happen at summer camp is we would have to do these trust falls. I don't know if you ever had to do one of these trust falls. And you kind of watch someone else go first and people all put their arms out and somebody has to fall back into them. And you're like, oh God, are they going to like catch them? And, you know, it's a really wonky analogy, but I think in many ways, therapeutic development is a little bit like a trust fall. And we are, especially in rare, dependent on investors to be investing in this space and sponsors to have guidelines, guardrails, and handrails that they can rely on so that we have predictability and consistency and transparency. And I think the real opportunity here with the Rare Disease Innovation Hub is that we're going to be able to trust that we're going to be able to fall into these spaces. And what I loved about the title of this workshop is that we're not just talking about all rare, we're talking about these diminishing populations that need even more trust. And I really loved the case study that was brought forward by the CF community that really outlined and articulated very well what a diminishing population is. And we have that in many places and spaces. And in many of our patient communities, that really just hit home what it means to be a diminishing population, what that feels like within the communities. And that can take on many forms. It can be because there are many clinical trials happening within the community. It can be because placebos aren't possible. It can be because traditionally required steroid naive patients to be in those trials, and steroids are now care standards within that community. There are a lot of different ways you can get to a diminishing population, but I love that this was the first topic selected. I do think we need to really dig down deeper and think about how existing guidances may not apply to diminishing populations, and what do we need to tailor those guidances and target it to those populations, because there are a lot of issues that don't apply to our diminishing populations in our rare disease communities that we need to better understand and dig into. The other thing I would just flag is that we had so many beautiful case studies from patient advocates and patient advocacy organizations, and that means that those organizations have been carrying the load. And there are so many rocks in the backpacks of patient advocacy organizations. And so as I watch that and I look at that, I really do wonder, how can we make that scalable? How do we ensure that we're not just always looking to the same case examples? And again, when we think about the smaller populations and those who are more and more getting left behind, how do we make sure that those who can't afford to be driving natural history studies and conducting patient preference studies are going to be benefiting from our learnings? And how do we facilitate that predictability and consistency for everyone? And then the last thing I would just say is that when I think about consistency, I think one of the greatest things FDA can offer us when we think about consistency is how do we take these discussions and these early learnings and again, share them with everyone so that, again, we translate that into a reliable trust fall. And that to me feels like guidance. And are there opportunities that have come from these conversations or other learnings that are now offerings that will start to take the shape of guidances? So.
Thanks, Annie. I love the visual analogy of a trust fall being required to bring innovation to patients, and that it really takes the hands of all the members of the community, from the drug developers to the patients, to the patient advocates, to the regulators, in order to make that happen. So thank you for that. Liz?
Hi, I'm coming from a history of being an investigator on rare disease trials since 2002 when we started in Fragile X syndrome and have subsequently moved to Angelman syndrome, Rett syndrome, Phelan-McDermott syndrome, Niemann-Pick type C, Batten's disease, and several others, as well as N of 1 trials. And I think that based on those populations that I'm treating, we don't we haven't really had the right mechanisms to do trials of the disease targeted and genetically targeted treatments that we've employed. And so I've heard three main points today. I think the most important words I heard were plausible mechanism pathway. And I think that's really important because it speaks to maybe a paradigm change. I mean, there's been some stuff written about early approval, but I think right now, historically in the past, we were treating symptoms, and those can actually improve in the short term, but now we're treating whole genetic diseases with a mechanism that's going to change the genetics of the disease. And we don't know how much we can get at what time within the disease we treat. And we need very long trials in general to understand that. And placebo standard randomized controlled trials can't go on for that period of time. So to answer the question, we need long-term studies. And so the proposal of an early approval based on some clinical signal, maybe not the same bar we would usually require, or maybe a biomarker, and then in a disease that's very devastating, and then earlier access with a very rigid requirement for a 10-year study with a protocol, something like that, where we can actually get information on how well is the drug working, because these diseases, the whole story is not going to be in a one-year study. This will provide access. It will alleviate the risk of not treating, and it will provide funding for the company because they have an approved drug where they can actually carry out the studies that will answer the question. And so I think this is tremendously important. And as an example, I'm going to refer to Angelman syndrome, where there were three phase 2 trials from 3 different companies with an antisense oligonucleotide that basically did the same thing, except it worked in different areas of the RNA. And all 3 of those studies showed benefit versus very well collected natural history with 600 patients on the Bailey, which is a performance-based measure, not just like a rating scale or something, as well as EEG biomarker change for delta power, which is linked strongly to cognition measures on the Bailey. So if you've got 3 independent studies showing the same thing on performance-based measures, what do we need to know now? Well, what we're doing now is 3 placebo-controlled trials that will tell us whether we have statistical significance versus a control group. But it's pretty convincing, right? So what we really need to know is, are we seeing everything we're going to see at a year of treatment? Or how long is that incremental rate of developmental change going to increment? And over 10 years, are we going to see something that's much more than we see over 1 year? Or does it work a certain amount and then taper off? And we can't answer that question with the current studies we're doing. So there's a need for a paradigm shift for these genetic treatments of rare genetic diseases to actually understand what's going to go on with these diseases. And I think what I heard in the plausible mechanism pathway is that maybe we're moving toward that, which I think is really important. The second point is within patient controls, or patient as their own controls. You can have a super rare disease and, okay, you can do a phase 3 trial that's 50 patients, and this is kind of a true story. And you can randomize 20 patients or 18 patients into the control arm. But these diseases are highly heterogeneous. So your control arm may or may not be the same as your treated arm. And so one study in the disease the control arm may have a faster rate of progression than ever seen before in any natural history study. Another randomized control trial, the control arm may have no progression at all. And then you're looking at data where probably drug approvals can be based on who is randomized into the control arm, so-called randomization roulette, rather than whether the drug actually has an effect. So we need other ways of measuring change. And someone mentioned, I think it was Marshall, mentioned that especially in degenerative disease, we're bringing patients in at different stages of degeneration. So the actual expectation is going to be different depending on what state of the disease we're in. And so I think it's the concept of within patient controls, or patient as their own control, where we can look at their pre-treatment rate of trajectory and post-treatment rate of trajectory is these are critical concepts, and that's actually a better control than we can actually find in an internal and potentially even an external control group. So I think that's a very important concept, and we really need to work on developing that. The RET gene therapies are going forward based on having good natural history and understanding what would be an adequate within patient control based on developmental milestones. And then natural history controls. We all agree these are tremendously important. Every single rare disease foundation wants to do this natural history study so they can have the data when they have a treatment. They call me, I talk to them, and they say, well, how many patients do we need? What do we need to do? How can we be like Duchenne's or CF or Angelman? And the truth is they've got $50,000, so they can't be like that. They don't have the ability to run those studies. So the question is, how much is good enough to at least have a little bit of ability to design trials and compare patients? And do we, to use those controls later on, do we need to have them in an FDA compliant database, which is humongously expensive, and none of these rare disease organizations can actually afford it? So I think better ways of understanding what's needed in natural history. What's the bare minimum for an ultra rare disease with a foundation that's trying to use garage sale money to collect the data that would be needed later on to help with comparators for treatment? I think definition of that is a really important agenda going forward.
Yeah, thanks, Liz. I think it's instructive that both you and Annie spoke to the importance of good direction from the agency in the form of formal guidance. I think that's a good parallel for both of your presentations here. Samia, your perspectives on the day so far?
Yeah, thank you, Steve. So as Steve mentioned, I'm Samuel Zaidi. I guess I'm here from as a representative from a gene therapy and perhaps an industry perspective. I do want to thank the organizers at Duke-Margolis. When they initially contacted me, I wasn't sure what I would contribute. Much of my work has been in external controls, but I'm very grateful for the opportunity. I do sit here as an Alexion Pharmaceuticals AstraZeneca rare disease employee, but one of the things that I've come to appreciate throughout the day is maybe something that I don't necessarily appreciate on a day-to-day basis, and that's the unique perspective that I may have starting in gene therapy about a decade and a half ago, and where we were in the early stages of the first in human for Zolgensma, where natural history was available and the opportunity to access natural history and witness a transformative therapy where the effect size is significant enough to drive the approvals. And even looking back then, survival, we were still looking into independent reviews of video recordings to ensure that the Bailey assessments and scores of the children sitting independently, video recordings of the children running around. And then over time, I've had the opportunity and privilege to work in less prevalent rare diseases where you witness therapeutics advancing at a faster rate than our understanding of the disease. And in these diseases, you may not have the luxury and the privilege of having natural history diseases. Some of the patients may not be offered or granted genetic testing until a therapy is in development or available. So the prevalence is not well recognized. You can't understand the disease. And at that point, defining endpoints or relying on a control is quite limiting. For some diseases, natural history is case series, literature, combining retrospective, prospective, and they may not always meet the regulatory compliance requirements for the data points. And I think we heard that throughout the day. We heard a lot about heterogeneity, working in lysosomal storage diseases, and we did hear from Kara earlier, neurocognitive. And when you do have that much heterogeneity, if you are to consider biomarkers or the stratification, you end up impacting power. I think circling back to what Annie referred to and Nicole mentioned earlier is the power of sharing the data and controls and control arms and building on that and partnering with regulatory agencies and the patient advocacy groups early on. From a genetic therapeutic or gene therapy, you're always dealing with the benefit risk profile and the irreversibility of the treatment that's being delivered to the patients. And I do think that when we're assessing that and looking at control arms, you're challenged with placebo studies, even in more prevalent diseases, because along with gene therapy, there are immunosuppressive regimens. So naturally, there's a higher likelihood of unblinding with the side effects of such regimens. Finally, I do believe that the early partnership in identifying what is meaningful to the patients and what would meet clinically meaningfulness as endpoints in the control groups, but also for your treatment arm is something that I think we heard a lot about early and often engagement.
Great. You did a great job of touching on a lot of the same points as your fellow panelists. And so as we move to Mark, I'll ask you what your takeaway from what you heard today is, but also perhaps if you have some insight into something you thought we might cover today that we haven't.
Well, thank you. Yes, I'm Mark Lemonson. I currently lead the Office of Biostatistics at CEDAR. I do want to thank the Rare Disease Hub led by Amy and the Duke-Margolis for putting on this very informative workshop today. I learned a lot, reinforced some of the things I already felt, and also learned some new things and continue to learn from my fellow panelists here. And I think focusing on controls is a great way to kick off these RISE workshops. I think it's somewhat overlooked sometimes in design. Naturally, I think we heard early in the day, it's part of the regulations to have a control, but it's more than regulations, it's good science. Attention up front should be given to this control, and will make our lives easier down the road. So I think this is a great start to these workshops. I would like to echo maybe as a little self-promotion what Dr. Prasad started with, that I have an office of 250 statisticians. Our reviewers are working very hard. You can be rest assured we are focused on these problems. Rare disease is one of the highest priorities in our office. People work hard to try to get this right. Maybe we ask a lot of questions, but we're just trying to get the best studies at the end to answer these questions, to provide effective therapeutics to sorely unmet needs. So we have several efforts within our office specifically for rare disease, and they're generally led by Becky Chu, who was on the panel this morning. We're generally divided by therapeutic area in our office, but the rare diseases span all therapeutic areas. So we have a work group where we exchange ideas and best practices to make sure we have the best thinking and most complete thinking for any particular problem. And we're trying to put more focus up front at what technically the IND stage or the design stage. That's where the bang for the buck is. We want to make sure these studies are informative. The worst outcome is to get to an end of a study with all the time patients have committed, the years of their lives, and all the resources put into it, is to have an uninformative study. I sit on a committee where each week we deal with studies that are hard to interpret. And really, the more effort we put up front in considering the controls, that's where the most value is here. Also, speaking of myself, I come from, most of my career at FDA has been in drug safety. And in drug safety, real world evidence is not secondary, it's sort of primary, for some reasons that it's so important in this setting in rare diseases. So I'm familiar, and I think my office is very familiar with the promises and challenges of real world evidence. And as Dr. Tidmarsh said in the opening remarks today, I'm glad we're going to make more use of that to supplement our trials. I think that's a very promising move in the right direction. We're also aware that we heard this today, and we're certainly aware that this is a community that will accept more risk in whether something is beneficial or not. There's no options. Generally, things are only getting worse. We understand that. guidances talk about regulatory flexibility, but this is not so much a concession. This is the appropriate action in this case. We have to account for the willingness of patients in the community to accept further uncertainty here. So that's perfect. That's well understood. This morning, we heard some discussion of archetypes. I do believe there are many different special cases, whether a treatment's going to be used once or chronically, whether it's expected to have an immediate effect or a long-term effect, whether it has to be used early in a disease progression or later. So there are many special cases, but I think thinking of kind of classes of them would be a very useful approach. And hopefully even provide successful examples within those classes, or at least consider what are the missing pieces to solve those different classes of examples. So I think that was a very good idea. Self-control, that's come up a lot. I wasn't even looking at the agenda, it wasn't clear that was going to come up at all. And I thought that was very important. So I'm glad that was repeatedly brought up. And there's many versions of that in different settings, but that doesn't remove randomization. Again, what Becky Chu talked about, we can do self-control studies with randomization. So I think that is a very promising area. And I think that will be maybe somewhat come into play in the next RISE workshop on individual care. Master protocols and hybrid designs. If we're going to have controls, and I recognize nobody wants to be in a control arm, let's make the most use of these controls. And that involves whether formal data sharing, structured master protocol, or as we heard in the previous session, the cystic fibrosis example, maybe that creates a lot of administrative and regulatory burden. There's other ways to share data in a master protocol-like way. But anyway, making the most use of controls we have. And hybrids, I think, just came up briefly. Again, hybrids will supplement the internal controls with external controls. So we're both able to evaluate whether the external controls are really representative of the study population, as well as gain power by increasing the sample size. So that's a very promising approach. I would like to say placebo is obviously kind of a bad word in this setting, but placebo doesn't necessarily mean no care. We don't mean it to be no care. People who participate in a trial should be getting state-of-the-art care. It should be almost an incentive to participate in a trial that they're going to get state-of-the-art care and monitoring. And we don't often know if these trials wouldn't be conducted if we knew the drug works. So it's not clear one arm is better than the other. So placebo has a bad name, but it's really versus the state-of-the-art standard of care here. I'll try to finish up a little bit here. Every patient matters, and that's not only because they're individuals and they matter, but with so few patients, we have to have as much data as we can get from patients. We have to find out what it takes by talking to patients and other means, what it takes to keep patients in the trial, how to design trials so patients stay in the trial and get the evaluations and measurements we need to evaluate the therapy. And I think it was only touched upon briefly, but digital health technology, to the extent that it's appropriate for a patient to wear continuous monitoring, that may be a very effective means to decrease burden on the patient, but continue to get the information we need for the trial. I'll be quick. Data quality. Before we get into any methods, I think we've heard, I'm glad this came out today. It doesn't always come out. Data quality is important. And right from the beginning, when we heard about natural history studies, the emphasis that these have to be designed prospectively and be as rigorous as trials. Because often we've seen external controls is, it's much lower quality data. The measurements aren't as rigorously taken or haphazardly taken. If we can get the same quality data from external controls that we expect in a trial, that would eliminate a lot of the need for having internal controls. I'll stop there. I may have some thoughts in other questions. So thank you.
Thanks, BJ.
First of all, I would like to thank the Rare Disease Hub and the Duke-Margolis for putting this excellent, informative workshop. I also want to thank all the participants, particularly the patient community and the advocacy groups, for sharing their lived experiences, and also the various subject matter experts for sharing their valuable insights. To me, the big takeaway from this meeting is that rare disease presents some unique challenges and opportunities, and there is no one-size-fits-all approach for study design when we look at the rare disease. We talked about lots of different options, including baseline controls, group sequential design, modified crossover. We also talked about the external control repository and multiple shots on the goal. So there are a multitude of ways to kind of conduct rare disease studies. Dr. Tidmarsh talked about the Commissioner's Priority Voucher, and Dr. Prasad talked about the biologic plausible mechanism. I want to expand a little bit more on it. So when we look at the totality of the evidence, it's based on the target product profile, biologic plausibility, size and consistency of the treatment effect, adequacy of the safety database. So those are some of the key considerations. And what the biologic or the plausible mechanism pathway also entails is how is the data collected and the evidence generated throughout the product life cycle. So one of the things that we would like to see more of is a balance between the pre-market studies and the post-approval studies of which data is collected when and how do we capture the most amount of data. Speaking of the data collection, my colleague, Dr. Najat Bushkush, talked about some of the challenges in the pediatric studies. Rare disease itself is challenging to study, and studying pediatric populations with rare diseases, in our experience, these challenges are logarithmically amplified, and there is a real burden on trial participation, both on the child and also on the caregivers and the families. So we would, to expedite the product development in the pediatric space, we would really encourage the product developers to explore alternate paradigms. And Dr. Bushkush mentioned about the E11 paradigm. So the prevailing paradigm is that we establish a proof of concept, we conduct some efficacy studies in adult population, then we move on to the pediatric studies with a PRIA PMR. What the new paradigm calls for is, if we have any available data, and obviously data should not be kind of wasted, as many of the panelists have pointed out, the study in the pediatrics should not exactly be a replica of the adult study. It should leverage any and all available data from the adult studies, and the pediatric study should focus on identifying the gaps that are there in the adult study and what studies can be done to address those gaps. The other issue that we did not kind of hear much about, but we heard it in brief mention, is that, again, we live in the 21st century. We have AI, we have digital health technologies. So how these could be harnessed to have decentralized trials to reduce the burden of study participation, enhance recruitment, and retain the study population. The last kind of thing that I want to also mention is that at the FDA, we will work collaboratively with the patient community, the product developers, we are open to reviewing any trial design that's backed by well-reasoned, scientifically sound rationale. And Amy Rick has been spearheading a lot of our initiatives to advance rare disease. And I just want to mention a few pilots that she's overseeing. We have the Start Disease pilot that's specifically for the rare disease. We have the RDR rare disease endpoint pilot that's also to advance the endpoints. And in the previous session, there was a brief discussion about harmonization. And within my center, CBER, we have the Cogent pilot, which I believe stands for Collaboration on Gene Therapies. This is a collaborative pilot with EU, Japan, and Canada. We have a few sponsors who are participating in all of these kind of like, pilot projects. We encourage the product developers to look at these pilot projects. Thank you.
Well, thank you all. That actually takes us to the end of our time here. So I'd like to thank our distinguished panelists. I'd like to thank our distinguished panelists. I'd like to thank Duke-Margolis for convening this meeting, for the FDA for sponsoring the meeting. And at this point, I'll turn it over to my colleague Garrett to help wrap up for the day. So thank you all. Thank you.
I guess we're going.
Time always does fly on days like this, but it particularly happened during that session. I will keep this very brief, noting the extensive wrap up that just happened about the day for the day. First, thank you so much, Steve. Final session participants, every contributor throughout the day. Normally, I'd probably highlight some of the big reflections for the day here, but knowing what a good job of that just occurred, and noting also that there's been, as promised, I guess this morning, some updates through the day that I think are probably going to be in all of our phones. Maybe I'll share just some brief highlights there and kind of put the challenge to us all to marry what's been discussed throughout the day with what's been announced here, and much more to follow or announced today. So a couple hours ago, FDA announced the Rare Disease Evidence Principles, it's RDEP program, to provide greater speed and predictability in the review of therapies intended to treat rare diseases in very small populations. I'm literally just reading from a press release. I won't go through it all, but a note, a few aspects that Dr. Tidmarsh, I guess, intimated earlier today. The RDEP is developed and implemented jointly by the Center for Drug Evaluation and Research and the Center for Biologics Evaluation and Research, so very much tied to the philosophy of the hub. Again, just touching on a couple aspects that I think we'll all look forward to digging into much more deeply, but it mentions that approvals under this process may be based on one adequate and well-controlled study plus robust confirmatory evidence, which may include strong mechanistic and biomarker evidence, evidence from relevant non-clinical models, clinical pharmacodynamic data, and the last bullet point touches on case reports, expanded access data, or natural history studies. I won't go any further into what I'm into it here. I think it's going to be the job of FDA and others, and then all of us to kind of see how we can marry all of this. I will say that I believe this is going to be distinct from what Dr. Prasad was mentioning on plausible mechanisms and getting additional details there forthcoming. So more to follow. I guess let us end with a second teaser then, brought up from this morning as well. So today was RISE 1 on internal and external controls. Lots of enthusiasm, clearly evidence through the day, the attendance, et cetera. But that's the first RISE workshop. Second RISE workshop, November 20th on individualized therapies. We have the same great teams on both sides diligently working on this already. And really we look forward to everyone's contributions there and the build-up, execution, follow-up, and again, just marrying this, all of this in action beyond just the meetings. To that point, well, a couple last logistics. Recording of the day's events will post in just a couple days. Workshop summary will follow later on. This is something we do for, I think, just about every Duke-Margolis FDA convening. And then, of course, my favorite part of the day always, beyond the great thanks to the speakers, panelists, moderators. A lot of work goes into the 4 to 6 or however many months leading up to today. Constant efforts between the FDA planning team, Amy Comstock-Rick, Pippa Friedman, Kathleen Davies, Nancy Guan, a couple of Duke-Margolis folks, and others in the background, by all means. A couple of Duke-Margolis folks that we cannot have any event without, Luke Durashar and Henna Vitello in the background for all events and communication type things. And then last but most important, this meeting cannot happen without the day in and day out, months-long efforts of 2 of my colleagues, Kristen Podsiad and Mia Williams. Thank you very much. Grateful for you all. Let's continue the work.